But before we do get started, I'd very much like to take a moment to reflect that we're all meeting on the traditional lands. And these are unceded lands across Australia. And this is something that we need to pause and reflect on every day as we appreciate where we are and the opportunities that we've been given through the generations and generations of connection that Aboriginal and Torres Strait Islander people have had in understanding this country, in believing in the country and their connection to country, seas, waters and skies. So we at the Kirby Institute, we're located on the lands of the Bidjigal people of the Eora Nation, and we'd like to acknowledge the traditional custodians of these lands and pay our respects to their elders past and present, and also extend those respects to any Aboriginal and Torres Strait Islander people joining us today in the rooms or who are joining us online. So we'll get started. I'm doing a very brief introduction, and then we've got like 3 or 4 sessions, and we are flipping and flopping between presentations, either in Melbourne or in Sydney. So hopefully we've got the technical stuff sorted now, but bear with us if there's a few glitches. So I don't think you need to go over this because we've been in the hub for about 5 years now. We were lucky enough to have an extension to the beginning of next year, or 2027. And the idea, of course, is for us to understand what are the commercial barriers in getting products such as improved diagnostics or new drugs or enhanced surveillance. How can we bring these together and accelerate their development to bring them to market? So the AMR Hub, and again, this is something that I think we're all familiar with, but is very much around that pre-commercialization phase, because it is a very complex, iterative process that involves a broad range of stakeholders to make this process work and come to fruition. So the AMR Hub is very much about that discovery, that space in between the discovery and getting something to market. So it's all around pre-commercialization of these products, trying to address the social and lab-based preclinical challenges. The idea is that all of this work that may have been done separately in all our different small silos comes together in a highly integrated way to increase those efficiencies and that cross-fertilization of knowledge. So we're supporting the development of new molecular diagnostic technologies and potential antibiotic compounds as well. And this is really overall with the goal of improving and advising on antimicrobial stewardship to transform these health outcomes globally. But of course, keeping in mind that for the purpose of this hub, in instance, we are using sexually transmitted bacteria as our exemplar. So again, the ASC funding, just as a reminder, is a little bit different to others in the academic world that may be more familiar with other sources of funding, such as NHMRC, MRFF, for example, where the focus might be on understanding the clinical outcomes or the benefits from a clinical perspective or a public health perspective. But this is very much focused on understanding what the industry barriers are to getting a product to commercialization. But again, recognizing that we can't do this alone. Industry need everyone else, and everyone else needs industry. So this is an opportunity for all of these multidisciplinary expertise to come together under the AMR Hub to try and address these challenges and barriers. A quick reminder of who's in the team, because many of us are we have our working groups, etc., and this is an opportunity to come together to remind us of who's involved. And there are a couple of new faces, particularly with the partner investigators, just to, for those who aren't lucky enough to have met Benjamin and Terry, for example, that attend our international collaborator meetings regularly from FIND and GuardP. They're the new representatives from these international not-for-profit organizations, and we meet with them frequently. So we had 2023, we all got together in one big hub, which was fantastic. And you'll see some faces here that are close to our hearts. 2024, we came together in hubs again. And this format was something that was suggested by the team as very much an opportunity to try and maximize the efficiency of way we're working together and to obviously minimize the expenses and to minimize our footprint as well. And it worked really well. So we're doing that again this year. Last year, we spoke more about the laboratory focus, accelerating antimicrobial stewardship, and you can see the speakers here, but there was very much a strong focus on the laboratory aspects and the pharmacological aspects. So if we take a step back to look at the different disciplines and expertise that are all part of this hub and all meet and constantly working together, this time we're focusing a bit more on the clinical, the social science, the health economics modelling and the epidemiology. So that's where the speakers and those perspectives that we're listening to a bit more today. So session one.
Yeah, no, thanks for the invitation, Tanya. And it's a little daunting following Cepheid as the global leader of diagnostics in this space, and then having SpeedX follow up as one of the great success stories of Australian manufacturing. So I don't know how I got the Guernsey, Tanya, but anyway, very happy to be part of this group. I'm a clinician. I would never have imagined that I was going to be starting a business, and I still see myself as a clinician. And so this part of my story is really about really early stage. What does starting a company look like? What do some of the challenges look like? Doing something different, moving from being a clinician into academia and then into business. But it's still a story of what motivates us at our core. And, that's, I guess, the intention of these images here is that in 2000, I took a journey to work in Kenya as a clinician in a remote rural hospital where it was the height of the HIV pandemic at that time. About 75% of the male and female medical wards were full of AIDS patients. We didn't have any HIV diagnostics. We didn't have antiretrovirals. We had kids coming in every single day with malaria and dying of malaria. We had 2 wards full of TB patients. And these things burned themselves into me and made a big impact on who I am and the rest of my professional journey. And it still continues in our journey as a company at Zip Diagnostics. So from that point, I sort of came home from that experience running clinical wards and emergency departments. Came back to do specialty training in infectious diseases in Melbourne, did a Master of Public Health to try and understand how we prevent disease and, stop that influx coming from upstream down to the clinicians downstream. And then did a PhD because I felt clinicians weren't very good at developing new tools. We're quite good at accumulating clinical evidence to guide clinical practice, but we don't generally bring our experience into generating new tools. So that was a great experience for me with James Beeson's group at WeHi at the time. And then I started a lab and was lab head at the Burnett for about 10 years. And that was a different experience again, so it's been a really interesting transition from clinician, public health, academia. And it's given me different perspectives on all of these issues that Tanya highlighted and Steve's also highlighted. But at the core, I'm still a clinician. I still go to the hospital every week and see patients, and it takes me back to patients and needs of patients and communities. And even though I'm in a tertiary hospital, I'm very much about community-based care as the principal driver for what we do at Zip Diagnostics and trying to provide alternatives to centralised laboratory-based testing, which I think is really core. And if we've got time, we'll touch on a few reasons why that is. So you can see here, there's a few examples of just the work I've done and my teams have done in these sort of countries. So that's a really important part of our story. So why start a company? I still ask myself this every day. It's a really difficult set of challenges from all of those other ones, and this evolution from clinical public health and academia into this new area has been a really interesting journey. I think it was because I was frustrated. I just was frustrated about not having the tools there, needing to make an impact, wanting to have an impact at scale. And I found that clinical work, I could see as many patients as I could see in a day. Academia, I could write another publication or another grant or develop a new tool in a lab, but I probably wasn't going to translate it into the real world in a big way that I want to try and do. So that's what Zip's going to try and do. I think we've still got a bit of a journey to do that, but we'll see how that sort of goes. So I guess, falling in a session with 2 amazing companies, I have to find some inspiration from somebody to start a company. And, who in this room would you sort of choose as being, the most amazing business person of all time, if you could model yourself and create a company? Could it be Elon Musk or I don't know who it would be, but for me, there's one standout, and it's obviously got to be Willy Wonka, because he is the greatest inventor of all time. And I just wanted to use this silly example actually to illustrate in a light way some of the challenges that we face starting a company. And I think Willy Wonka had a lot of them sorted out, so I'm pretty happy to sort of follow in his footsteps to some degree. Now, there's too many slides. I won't go through all of them. If anyone wants to talk about any elements of this in detail, come and talk to me later or email me. But I think one of the first ones is you've got to have some imagination. I love this scene where they open up in Willy Wonka and the Chocolate Factory. They sort of step into this dream world, and you just see the vision, the creativity of this person that's driving this company. But what's different is the dream that he's living is real. Anything can be picked off and used, and it's real. And I think that's a very important difference between dreaming and wishing for something, but actually having a dream and an intent that is going to translate into something. And we have to keep that dream and true intent at the core of what we do. So that's what Zip's mission is, basically to create point-of-care diagnostic tests that are rapid, accurate, that they are low cost, that they can be deployed in true point of care. I want to see them next to healthcare professionals. I want to see them in community clinics. I want to see them in remote areas, not just stacked up in laboratories or fast throughput tests in specialised clinics. So I think of it in 4 main domains: point of care, genuine point of care, fast results, having high performance, quality, trust of the users. It's about price, but it's more than price. A lot of people ask, how much is your test going to cost? It's all about really health economics and the value that the test brings, which is much more than just the price of consumable. And it's about production. And I think we're hearing more about Australian manufacturing, but Australia does not make enough stuff in our country, and we have to change that. It's a really important thing that we're losing a huge skill base, and we're not exploiting our scientific expertise enough because we don't make enough stuff in Australia. So these are sort of core parts of it. If you're starting a company, you've got to find people you can talk to. It can be a bit lonely sometimes, sort of starting a company and being a founder, and you need people that you can share the journey with. And there's highs, but there's lots of difficulties too. And so for me, this was Bill Hopper, who's the co-founder of Zip Diagnostics with me, and he brought a lot of expertise in engineering and manufacturing. But, we just bring different perspectives to problems, clinical and public health and assay development, manufacturing and engineering background. And that's really rewarding for both of us on a personal level. And it's really important for the company. Clearly, Willy Wonka also had great products, and at the core of a manufacturing company, a diagnostics company, it's got to be about your product. And we've sort of evolved out of an initial focus in healthcare for human applications into some other domains like animal health, production and companion animals, and environmental testing, because we're seeing exactly the same problems and needs in those domains as we see in human health. So, whether you're caring for animals or you're doing water testing or air testing, you need data-driven decision-making wherever you are in those remote areas. So that's what we're sort of getting into a little bit more. We've basically made a very strong commitment to instrumented systems. A lot of people try and run away from equipment and say, oh, I can stick a thing in my pocket and read it by eye. That's going to be a really cheap test. I fundamentally see huge value in instrumentation in terms of quality, managing workflow, training, and at the end of the day, a test is just data. So for me, it's all about a data environment and data management and data security, and instrumentation really gives us that ability. So these instruments that we've been developing, the top two are isothermal nucleic acid amplification systems. The first one of those is approved for use in Australia and in Europe and the UK. The middle one is one that we've got in development that's a high multiplexing sort of instrument. And then the bottom one is more of it's like a lateral flow rat type test reader, but the difference being that it actually has multi-analyte array capability, so we can actually get a lot more out of the standard sort of lateral flow tests that you see a lot of. Just some quick examples. So this is a trip that George, who's one of the postdocs with me, we did a trip to Malawi, and you can sort of just see how genuinely portable this system is. We literally carry it out, in a small case, set it up with a battery, whichever clinic we happen to get to. So that's really nice to be able to get into remote areas and just set up literally in about 30 seconds. This is some testing that we did just a few weeks ago in Western Victoria, where this dairy farm was having abortions in the cattle, and they were not quite sure what that was due to. So we set up and brought one of the first mobs through, 100 head of cattle, and by the end of the day, we could tell them that 80% of the cattle actually had a disease called pestivirus, and went back again another 2 weeks later to see which ones were chronically infected. And so that became really valuable information to that farm to identify chronically infected cattle that were continuing to infect the rest of the herds. This is another example of some exotic diseases. These are diseases that we don't have in Australia, like foot and mouth disease, lumpy skin disease, that are through Indonesia and the Asia Pacific region. They would cause devastating impact on the Australian industry if they got into Australia, probably in the order of $6 or $7 billion a year. So this is trying to get some of these portable tests into remote farm communities in the Asia Pacific region. This is another area we're getting into at the moment with interesting environmental testing. Obviously, we have great infrastructure in places like Australia with sewer systems and septic tanks and things like that. But even in Australia and high income countries, we see failure of the infrastructure. And so we get leakage of sewers and septics into our water systems, our rivers and streams, and out into the beaches where we swim. And we're really interested to try and help identify those breaches in infrastructure so that we can actually continue to improve and keep our environment safe and healthy for us and all the animals that we share it with. And on the right-hand side, you can sort of see there's some maps there which really get at that issue that I was talking about before, which is around data and data systems, including incorporating test results into geospatial data so that we can actually, in real time, map where this disease is exactly, and that helps us to tailor our response with greater precision and effectiveness. This is just I don't know if you remember this scene from the movie, but yeah, there was a scene where they go into the inventing room, and you can see the craziness behind Willy Wonka and the way that he puts things together. But I think for me, this is sort of a really important question for our scientific ecosystem. We're very driven by discovery in Australia. We're very driven by technology. And I think that's right. We need that. But we need a better balance between translational research, but also addressing problems for what they are and bringing in a multitude of solutions that usually involve multiple technologies, because the solutions are complex and they actually need multiple elements to fix them. And so that's why I call this inventing mindset as opposed to a discovery mindset. And it's quite a different way of thinking, and you need quite different teams. And that's what we've also tried to build in SIP. So yeah, just making the point again here. So discovery is, I find something new, I discover something new, and I work out why it works and how it functions. Invention starts for me as a problem, and how am I going to solve this problem? And I'll go to any tool that I need to try and solve that problem and create it in an integrated and usable system. So for us as a young company, discovery and invention sort of takes different cycles, but much more structured in the way that we design new tools, processes, procedures that we have to write and follow, and also very quick iterations of prototyping and testing. So the faster you can build something and test it and iterate the design, the more successful you're likely to be. And so that's very much what we've done at Zip is build our own engineering department as well, so that we're building our own prototypes and moving very, very quickly to develop new things. And that's the same for our assay work as well. Of course, you remember the chocolate river. No one else mixes their chocolate by waterfall, but if it's the only way if you want it just right. So we also had to build a lot of facilities for ourselves at Zip. So we had to, during the middle of the pandemic, build our own PC2 lab, ISO 7 clean rooms, and now engineering department. So a lot of our effort in the last 6 years has really been in infrastructure. And a lot of people don't sort of understand this huge investment that goes into manufacturing. It's not the same as having a R&D PC2 lab. It's actually quite different processes that you have to follow to get these infrastructure in place. And a broad set of capabilities for everything from bioinformatics, recombinant protein expression, assay development, lyophilization, all of these really critical. Team and culture, really critical. And as a small Australian manufacturer, we have to draw on the talent that we've got at our fingertips. And as a small company, you have to draw on the talent that you can afford. So you've got limited resources. And this is where I'd really love to see more academia coming into industry settings, having an industry experience, really see for yourselves what it's like. The R&D that we do in an industry setting is just as exciting and innovative as it is in any university, but you'll learn a lot of other skills as well. And learning quality systems and translational commercialization sort of elements will be, I hope, interesting and useful for future careers.
So thank you, Jack. I really appreciate it, because I think just to recognise that Jack was invited, particularly by Shivani, to join us as a guest to bring this perspective of a company that has an Australian company, obviously has a broad basis of the technologies that they provide, and also that with respect to that sovereignty along the entire pathway of that manufacturing process. So that's at least the principles and the goals of the company. So thank you so much, Jack. So Nicole, I introduced herself before, but I think the great thing about this perspective, obviously, SpeedX are a massive supporter and hub partner, and without their support, this hub wouldn't have been possible. But I just wanted to give Nicole the opportunity to reflect on the perspective of an Australian company here and why they got involved in the hub. So I think that was the remit that we spoke about, Nicole. So I'll hand over to Nicole.
Hello, thanks for having me. After being invited for this talk, I realized I've actually never done a talk other than talking about insignia. So this is a little bit nerve-wracking, but hopefully I'll be able to give you some insights into the hub and how it has helped to accelerate diagnostics in our company. So SpeedX spun out of a multinational company, Johnson & Johnson. It was built on foundation IP, co-invented by Alison Todd and Elisa McCarney. They are the co-founders of the company and essentially the heart and soul, what keeps it going. SpeedX was created in 2009. It started as a small startup, licensing the impressive IP that we have and custom designing assays for other big companies. It has evolved over time and it turned into a more mature company, creating its own molecular diagnostic tests and being able to stand next to, competing with, and collaborating with big companies. Following the boom and gloom that COVID brought to the molecular diagnostic world, SpeedX had a little change and brought in a new CEO, Jeremy, who's there on the slide, who brings a wealth of knowledge in diagnostics. So after 16 years of being a company, the future still looks bright for SpeedX.
Go to that one?
Yes.
Okay. Sorry about that, guys.
To give you a little more snapshot into the company, we have some amazing technology that powers all our products. We've managed to actually generate quite a large inventory of patents, a total of 160 and 125 trademarks, along with having on the market more than 50 products. The technologies invented include Plex PCR, which is the detection technology that enables us to swiftly multiplex. Our Plex Plus technology, which is the latest invention, it further enhances the multiplexing capacity that we have, doubling results outputs and increasing throughput. Our Plex Prime technology, powering SNP detection and used in our Resistance Plus products. And Insignia, a novel RNA expression analysis method used for viability assays. I think at the moment we have about 50 or so employees. I don't know the exact number. And these are all spread within 3 different sites. So we have our headquarters in Sydney, but also offices in the US and in London as well. We have partnered with many organizations and companies, Cepheid included, having our Resistance Plus GC assay on their Gene Expert platform, and many others out there. The mission for the company has always been to deliver clinical diagnostics to improve patient outcomes. So rapid product creation is something that we can easily achieve, or relatively easily. And that happens with the combined experience that we have within the team, from innovation, R&D, regulatory, manufacturing, sales, and marketing. We've got the research know-how using the technologies that we mentioned before and leading the way in multiplexing. We've launched products for IVD and research use only, and have the backing of clinical and regulatory experience. The development and manufacturing all happens in-house. It starts from strands of DNA to a neat little box. We have knowledge of the market, commercializing kits since 2016, both to direct customers and indirectly through our partners. And we've also been able to influence changes in the field and create new markets. But it is our collaborative partnerships that support us, with collaborations leading to launch products. The diversity in our network is what makes this happen. Similar to raising kids, biotech takes a village. And this is true for the whole product lifecycle, starting from the idea, the need for something, to making that happen, building the blocks for our assays, going into clinical and regulatory fields. And all of this is done with support. That's how it's been from the very conception of SpeedX as a company. Our journey and our product lifecycle involves engaging with every sector of our deeply interconnected biotech ecosystem. 2 of our pioneering products in the world of resistance-guided therapy originated this way, through collaborations with individuals who are also part of the hub. The example there is gonorrhea, our gyrase SNP detection, ruling in ciprofloxacin resistance, ruling in ciprofloxacin treatment for cases where individuals are susceptible. A product that was developed closely with the University of Queensland and David Wiley's group. As well as the OG, our Mycoplasma genitalium test for macrolide resistance, which significantly improved cure rates. This originated from an idea from Cepheid and was championed and propelled forward by Kat and her team. These 2 products further solidified the power of collaboration. So to take it to the next level, we had to be a part of the AMR Hub. The Hub not only brings together the collaborators we already work with, but it has expanded the network beyond what we ever could have done on our own. It has consolidated and focused the efforts, truly creating additional opportunities and pathways to enable faster generation of data to support commercialization of a product. From the perspective of a diagnostic developer, it opens up opportunities. But as Tanya mentioned at the very beginning, it also works the other way. Now, I think I have way too many clicks on here. Now I have to use the example of Insignia to demonstrate how this can be achieved with the Hub. Insignia is a novel viability assay, and we're essentially trying to generate change where potentially individuals will be told that they don't need treatment for non-viable infections. It's hard work. And we have first-hand experience in how the Hub has accelerated the progress in generating clinical evidence for this. So we can certainly tick off all the points that are included here on the slide. For health economics, Ben, Hugh, and his team are contributing to building the modelling and evidence to demonstrate how much antibiotic use could be saved if viability assays are introduced to the market. We have discussed working on understanding whether or not individuals would be open or not to not being treated if viability assays were used to diagnose their infection. We have been able to access clinical samples, for example, receiving hundreds and hundreds oropharyngeal and saliva samples from Shivani and her team, all from the omega-3 study done with Eric Chow at Melbourne Sexual Health. And this has helped us further develop our assay and test iterations of it along the way. It has also helped us see how this performs against gold standard methods. We've successfully carried out prospective clinical studies with Arthur, who's in the room, at Sydney Sexual Health, in order to test the feasibility of not only the viability, but also the AST component of the assay. The protocol for that study also arises from contributions from Kat Bradshaw and Tanya and Rebecca Guy, also supported with additional Hub funding. And all of you have been instrumental in making this a reality, your intellectual contribution and vision. Regarding funding, Tanya Applegate led the team in the submission of a successful Ideas Grant application funded by NHMRC, and with Insignia as the focus, for which we'll be starting a clinical study soon and analysing the performance of the assay again. We have discussed and showcased our assay to international health organisations to aid in starting the conversations regarding implementation. And publications are out there supporting the use of our test like others available, and other manuscripts are currently being written. Data from the research that has been carried out has been presented at multiple conferences worldwide and being really well received in the STI community. All of this is building the case, generating the evidence and the need for a test like Insignia required to demonstrate or to show that there will be commercial success, uptake, and ultimately will support the use of the assay in the wild. It has taken a village, but we have progressed so much further than we could have on our own. So I'd like to leave you with a quote by Helen Keller, an inspiring figure in American history, who expressed her belief in the power of collaboration. Alone, we can do so little. Together, we can do so much. Thank you.
Thank you so much. So I think that perspective from my view is also a great example of what's possible when the co-founders are still part of the original company, driving, making decisions, keeping the principles and the core foundations of what's the purpose of why the company was developed in the first place, similarly to as Jack was mentioning, which is a feature often of sometimes maybe smaller companies, but when you've got that corporate knowledge from the beginning through and continuing through, I really feel it does make that ability to see through the weeds. So we are well over time, thanks to the fact that we have an incredible audience that were welcomed in the beginning as well. So I think we do have time for 5 minutes of key questions.
Thank you to the three amazing speakers. I have many questions, but I'll focus on one. I'm just interested in terms of the diagnostic manufacturing side in Australia and the trickiness that maybe the costs that companies face in terms of trying to keep the sovereignty of manufacturing in Australia. I wonder whether any Nicole or
I guess
SpeedX or Zach,
or
Jack might want to comment on that.
Maybe Ali or Elisa equally, because this is obviously a massive consideration Australia-wide across all manufacturing, right? And particularly in diagnostics. So actually, I'll just throw it to Elisa for the moment and then quickly to Jack.
I mean, definitely there is an expense to keeping it in Australia, more so than looking international at some of the markets that can do it cheaper. But also I think it's about control and quality control and how much of that system you can actually manage yourself. So I think maybe Jack's perspective with manufacturing a device could be very different to us when we're manufacturing kits. We have components we assemble in Australia, but those can be manufactured in other countries. Obviously, you could probably all set up a site elsewhere, and we had looked at it in the past. But ideally, I think when you can keep it closer to home, there's more control of what happens and the regulatory systems. It's not to say if we expand hugely, we do have sites we've looked at where we could upscale to significant levels and have that in place, and that probably may not be in Australia.
No, it's a great point around the sort of supply chain and control over the entire supply chain, because we've seen so many examples of that, which has sort of fallen apart along the way. So Jack, would you like to comment on that, understanding you have a different perspective from even some of the proteins that are made internally for your assays and the diagnostic device itself?
Yeah,
What are the challenges of keeping it in Australia?
Yeah, I mean, we started life wanting to be the manufacturer. So that was clear in our intent in forming the company. So I think that's important because not every company will have that intent. And we wanted to be manufacturing as much in-house as we could, so be end to end. So that's currently what we do. It's required then, the only thing that we really buy in is actually primers and probes, and it's actually almost impossible to get any of those made in Australia. It all comes from China, US and South Korea. And that's something that we think should change in Australia as well, because it leaves us highly vulnerable to timelines and accessibility to those really key reagents. But we make all of the proteins that go into our assays, for instance, reverse transcriptases, DNA polymerases, do all of the assay formulation, lyophilization. We actually build our own machinery for manufacturing automation, and that gives us our scale-up capability. So that's for all of the assays and then the instrumentation side of things too. But, you know, the point was just made before, it's not just about the manufacturing and the scale of manufacturing, it's the quality system that all of that has to happen in. And that is incredibly demanding and was something I was completely naive to when we started the company. But we grow into it, and it's so important. Like you have to make thousands and thousands of the same thing reproducibly, and it has to work every single time when it gets to a patient. And you need a quality system to drive all of that. So that means your staff training, every piece of equipment is calibrated, every procedure is written and authorised. data management, every element of quality goes behind all of these systems. And so sometimes people think, oh, why does it cost so much, to get things made by companies? And that's why it's largely about that requirement. And you can't shortcut that. You shouldn't shortcut it either. So.
Thanks, Jack. It's a really important point you raise around the cost, the perceptions of what it takes to cost, because there's obviously always that pressure. And I know Cepheid, Steve, you would be one to comment on this with regards to what it takes for the development, but then obviously what is possible in terms of cost to get it at scale to people in need. And then clearly that balance between high income countries that can maybe able to afford to pay versus lower resource settings where the capacity to pay is an ability to pay is much lower in terms of affordability for those settings. So I just have one quick question to bring Steve into the conversation, if that's okay, Steve, if you can hear me okay. Because they were perspectives from Australian companies here and the challenges of keeping things in-house and at that quality. I was wondering from an international company's perspective, this is a big question, and we don't have long to answer it, unfortunately. How do you figure out when it's worthwhile going into a country? So, for example, in Australia, with regards to the market entry, why is it worthwhile for you to come into a country with a small population? The cost of regulations are high, the timelines are long, as opposed to focusing elsewhere? Because I know this is a conversation we have all the time around international companies again. Why would we bother coming to a country like Australia? Tell me why we should bother. Being a large company, I imagine, I'm just guessing, I'm assuming that you have some resources around doing that market analysis. But smaller companies, how do they manage that market analysis to determine that there's a viable market in a place such as Australia? I'm just using that as an example, where the priority populations might be small, the profitability might be small, but the medical need and the health equity demands that these tests are made available in these communities. Big question. Sorry to ask you to answer it in 2 minutes.
Yeah, I can probably keep it to one.
Okay.
I think the key here is around some of the central tenets that actually drive, the research. In some ways, it's not that different. So it's about, will the test be acceptable? Is it feasible in that environment? Does it have clinical utility? And will it be cost effective? And I suppose, all manufacturers at some point are looking for a return on their investment. And I think at the end of the day, whilst issues around, being able to drive down pathogenesis of different kinds, ultimately it's got to be at some point about cost recovery. But then it's also about its potential uptake and value in that particular environment. The other key thing too is that it's not always just about investing in, the selling of tests. For example, Cepheid has invested very heavily in Australia and Australian researchers because of the evidence they generate and also the impact that evidence has. So I think there are a couple of other key pieces. It's not just a commercial decision or it's not driven just around marketing. It's also about partnership and the value of that partnership and the two-way value of that partnership and what that brings into the space. So in a snapshot, that would be my reply, Tanya.
Thanks, Steve. I think you've touched on a critical point, which we've been fortunate enough to have people in the companies that we've worked with that recognise the value of those partnerships and how long they can take and the trust in those partnerships to establish them, particularly. But in other organisations, establishing those relationships can take a long time, and being able to find the right people that understand the value of these long-term academic, government, and industry partnerships can take a bit of while and time to establish. So I just think we can't undervalue that time and investment in establishing those relationships in the first place. So I think we're going to have to.
One final point too, Tanya.
Yeah, go for it.
Is that because we're a long way from nowhere, making ourselves visible is the other really key element. Australia is a leader, in diagnostic innovation and a range of other public health outcomes. And it's often one of the bigger challenges is getting that innovation in front of people and getting them to hear the value when it's such a crowded space anyway. But that's one of the reasons why Australia leads the world in a number of different areas, and particularly in terms of the evidence it generates. It's a matter of who you get that in front of and what value it brings to the table in terms of whatever you're trying to negotiate.
Thank you. Australians aren't traditionally very good at putting themselves in front of everyone and just saying, look at me, but it's something that we're working on.
Cool. So, hi everyone, I'm Shiva and I'm a postdoctoral research fellow at the Sydney Centre for Healthy Societies, and I'll be chairing this session on co-design in antimicrobial stewardship programs. So I'd like to start by acknowledging the traditional owners of the lands on which we meet, the Bidjigal people, and pay my respect to elders past and present. So interventions, technologies, and tests are produced because of social arrangements. So think about funding models. But they also exist in relation to populations and individuals and take on meaning and relevance in the context of their lives. So to think through this today, we have 4 speakers. I'm one of them. I will present on GBTQ perceptions of urgency as it relates to antimicrobial resistant STIs. Then we have Dr. Arthur Wong, who will present on antimicrobial consumption in men who have sex with men. Dr. Terrelyn Ludwig will present on AMR and asymptomatic STI screening. And finally, Mr. Matthew Vaughan will talk to us about reflections on DoxyPEP from a community organization perspective. So I am going to get right into it and start timing myself. So this presentation is based on a project in which my team and I are exploring how gay and bisexual men and trans and gender diverse people in Australia view and understand antibiotic-resistant STIs. So that's all of us there. So within the broader context of AMR, we know that health agencies, governments, and practitioners have issued increasing alerts about the rise of antibiotic-resistant STIs. So we all know infections of high priority, as identified by experts, are gonorrhea and Mycoplasma genitalium because of their resistance profiles and consequent treatment challenges. So the WHO has identified gonorrhea as a pathogen of high priority, and the Centers for Disease Control and Prevention has added M. genitalium to its watch list of resistant microbial threats. Right, but the idea of urgency, research and AMR advocacy often starts at this point, right? With the a priori assumption that the urgency is collectively agreed on, and it's accepted that the crisis has arrived. However, this context privileges the worldview of experts as the starting point. In such contexts, decisions have been made by agencies, governments, scientists, academics, and others about what is pressing and why, right? But what about everyday people? So in this presentation, I inverse the framing of urgency from something that is top-down to consider what it would mean from below. So what would make antibiotic-resistant STIs feel urgent to people on the ground? I think given the fact that the population of focus in this study is GBTQ plus people, it's important to flag that historically, this, creating a sense of urgency from below is not foreign to this population. So thinking back to the HIV/AIDS crisis in the face of government lethargy, especially in the US, it was these populations that created and pushed for better responses from below. So this dynamic's not new. So who did we talk to? So this study is based on semi-structured interviews with 49 gay and bisexual men and trans and gender diverse people in Australia. Of our 49 interviewees, 43 were recorded male at birth and 6 were recorded female at birth, and 11 people identified with a gender other than the one that were recorded at birth. So most participants identified as gay or bisexual, and others identified with alternate sexualities and or combinations of sexualities. Ages ranged between 19 and 61. 34 participants were born in Australia and 15 were born overseas. And participants came from all Australian states and territories spread across urban, rural, and regional locations, which becomes important when we start thinking about urgency. And it had a nice spread of different ethnic backgrounds as well. And obviously all the names I use in this presentation are pseudonyms. So first thing, what do people know? So it might sound common sense, but a key on whether resistant STIs felt urgent was what people knew about them. Some participants explained they did not know much, whilst others demonstrated a deeper grasp on the topic. So for example, Miles said, I don't know anything about antibiotic-resistant STIs at all. I didn't really know that was a thing. People who had an understanding about resistant STIs often spoke about gonorrhea as being the infection of concern, and then other people mentioned things like, oh, chlamydia and maybe syphilis to a lesser degree. So for example, Jackson said, I don't know the subtype to the strains, but in terms of like ceftriaxone resistance, there's a big problem. And also there's known gonorrhea types that are resistant to everything. So they do exist. So yeah, it's a big problem. However, many participants had no knowledge about M. genitalium, right? So they didn't know it existed, let alone the high rates of antibiotic resistance that can make it especially difficult to treat at times. So if people were aware of M. genitalium, it was because they had the infection at some point or knew someone like a friend who had tested positive for the infection. And I think it's because we don't perform quarterly testing for this infection, right? So gonorrhea, chlamydia, syphilis, and HIV, you go every 3 months to get your prep script, you get tested for it, so you know they exist. So with M. genitalium, because that doesn't happen, it's kind of not part of the imaginary of something that exists, yet alone could be resistant, so you can't really be concerned about it. Optimism was something else, right? So feelings of optimism, this idea that things were going to be okay, tempered the extent to which resistant STIs felt like a concern. So to some degree, this was shaped by recent medical and public health successes. So for example, Miles said, because the two examples of COVID and monkeypox that relate closely to me, as we both ended up with a vaccine, thankfully, once it got really serious. So just be hoping that sort of gets solved before it gets to a really worrying sort of occurrence. So crucially, such optimism has implications for like the temporal dimensions of resistant STIs by imbuing the future with a sense of hopefulness. Personal circumstances were also important. So the sense of, so the urgency and importance of resistant STIs was also shaped by its relative importance to other life circumstances, such as making ends meet. So Hamish explained, concerns about resistant STIs are at the bottom of the barrel. At the bottom of the barrel, unfortunately, I'm struggling to keep myself afloat at the moment. I don't have time to worry about that right now. So the vicissitudes of just everyday living and struggling to get by centers the importance of the present in people's lives, right? That leaves very little space for something like resistant STIs to feel important and the here and now. So it gets relegated to something for the future. So the geographic dimensions are really interesting. And I guess this was a great thing about talking to people all across Australia. So it really shaped some of the ways in which urgency was felt. It informed one's sense of assumed proximity to resistant infections, including both national and international dimensions. So for some participants, resistant STIs were more of an international concern, right? So Roman said, as far as I know, gonorrhea is the only one that currently has where there's a real issue or the risk of an antibiotic-resistant strain. And that antibiotic-resistant strain, as far as I know, is still relatively rare in Australia. So it's not like I'm freaking out about it. I'm a little bit worried about it. Infections such as COVID-19 and the outbreak of Mpox in 2022 served as sense-making devices used to imagine possibilities of how antibiotic-resistant STIs might spread geographically. So Luke said, because it's like when monkeypox was here in Australia in 2022. So you were getting cases in like Sydney and Melbourne. There were none in Tasmania. The same with COVID as well. The bigger cities seem to get these things first. So major metropolitan Australian cities like Sydney and Melbourne were conceptualized as these hotspots. The infections were seen as something that would spread from these regions, and this infection creep tempered the perceived risk of contracting resistant STIs, which meant that in people's minds, right, geographical location was a protective factor, which they were thinking about in these potential futures where we might be getting more resistant strains of STI spreading and the sense of urgency around them. This was really so participants' considerations about the prevalence of resistant STIs was a critical frame through which they thought about the urgency around it. So Archie said, if I used Mpox as an example, that did change my behavior quite substantially, where I started thinking, no, I don't want to fuck with anyone until I kind of learn more about Mpox, understand what the risks are, understand transmission and treatment, and kind of what the local prevalence is. It would probably be something similar. So prevalence thinking introduces, an important temporal dimension to the urgency around resistant STIs. It orients urgency to the future by framing it in terms of escalations that are yet to come. Importantly, the emphasis placed on prevalence exists in a dialectical relationship with other factors that mediate the sense of urgency surrounding resistant STIs. So, for example, Julian, he knew someone who had contracted a resistant strain of M. genitalium that took about 6 months to get treated to clear the infection, and he spoke with this person. However, Julian was not too concerned about resistant STIs, as there hasn't been some sort of explosion in the community that would cause me to drastically change my attitude. He said, it was a bit of a concern about it at the time, but in my brain, it was just quarantined to 2 people who were not fooling around with other people. So it wasn't a concern, but yeah, that's probably limited thinking. So Julian's reflections illustrate that the interplay of proximity and prevalence, right? So the resistance was proximate. He knew someone who had it, but it wasn't prevalent. So the numbers were minimal. And it was this relationship that meant it wasn't a big concern. So what does all of this mean, right? Essentially, it means that urgency about resistant STIs from a community perspective is a relational and multidimensional mediation. The way it means that it's produced at the intersections of people's subjectivities, their life circumstances, feelings of optimism, knowledge about resistant STIs, pleasure, histories, and past infection outbreaks, which act as sense-making devices, alongside considerations about bacterial proximity, bodily integrity, and potential risks. So why does this all matter? It tells us that community perceptions of urgency may not align with expert sensors of urgency. It is important for working with communities, as it means not assuming that everyone thinks about crisis in the same way or even knows much about resistance necessarily. It also means that when you communicate with community members, you should be doing so with an understanding that people may not be thinking like you and are making complex considerations about how to navigate STIs, and that this is not one-dimensional linear thinking, right? And this has implications for how we frame and communicate technologies to communities, as we need to think about how these technologies will fit into different lives and where they can work best. I think what it also tells us is that there's scope to think with community members about the complexity of the unfolding situation and to engage with them in the complexity of it as they are already thinking about navigating resistance in interesting and insightful ways. So when we think about new technologies and interventions, we need to do so with an understanding that at least for, LGBTQ plus populations, they're a pretty savvy population that has complex ways of negotiating pleasure and STIs, which I think has to do with HIV/AIDS and the historical legacies of that. It also means potentially framing new technologies and interventions in terms of things like prevalence, as communities are already thinking about AMR in this way. It's already a framework, so why not use it? And finally, the optimism means that about solutions, right? Or the future means that I think this is a population that is receptive to science and technology. And I think they believe in the ability of the work that, is happening in industry, for example. I think they believe it can make a difference in their lives and they would be open to integrating it, I don't think you'd be working with reticence and coming up against that. I think people are very happy to embed it in the complex lives that they live, considering the factors that I've just spoken to you all about. That is all from me. And I will now pass on to Terrelyn Ludwig, who will be joining us from Melbourne for the Sydney people here.
It's Arthur.
I lie. It's Arthur. Arthur's next.
Fine.
Cool.
Because I want Arthur to do the introduction so I can save some time.
Yeah. Hi, hope everyone can hear and see me. I'll just see. Great, the slides work. Okay, cool. Okay, my timer is on. Hi everyone. I'm Arthur. I'm a sexual health clinician working in Sydney, and hopefully will be a card-carrying epidemiologist once I get my PhD. And the title of my talk is Are we giving too much unnecessary antibiotics for chlamydia and gonorrhea in cisgender men who have sex with men? Disclosure, big fan of the Hub. I receive a PhD scholarship from the Hub, and all the work that I'm about to present have been supported by the Hub, and also most of them have substantial contribution from co-investigators. So just a really quick intro about where I work. I work in Sydney, New South Wales, and like many jurisdictions in Australia, we continue to have really high notification rates for chlamydia and gonorrhea. I'll focus more on gonorrhea, which is my favorite STI. Gonorrhea continues to rise, and it has a huge kind of disproportionate notification rate for men, driven mainly by MSM transmission. And this is our state's STI strategy. And unsurprisingly, because of the notifications, MSM remains a priority population for kind of offering kind of testing and treatment paradigms for chlamydia gonorrhea, but also where I work, a publicly funded sexual health clinic, is a priority setting for doing that. What I'm about to say is probably really basic information for this audience, but just for the sake of the talk, we broadly think about strategies to test and treat gonorrhea in two ways. This is a simplification. So someone like me may see a patient in clinic who have symptoms that may be due to chlamydia and or gonorrhea. I see them. I sometimes take a history and do an examination with or without microscopy, and we determine the likelihood that someone has these infections. I offer them some preemptive antibiotics to hopefully cover for these bugs and get them feeling better. But at the same time, I would usually send some tests to confirm whether or not they have these infections. And they're usually very sensitive nucleic acid tests that are done in centralized labs. Alternatively, we also encourage subgroups of people without symptoms, such as MSM on PrEP, to come to the clinic regularly to do asymptomatic screening for chlamydia and gonorrhea in a paradigm we call 3x3 testing. So every 3 months, they give 3 anatomical site testing, so in the throat, in the rectum, and in the urine. And if they test positive for these infections like gonorrhea, then we give them the appropriate antibiotics according to guidelines. But this is obviously a huge simplification. I haven't included situations like if they were contact or if they wanted to get testing before treatment for symptoms, or if you work in a setting with molecular point of care available. But I think it doesn't take a lot of imagination to see that in both of these pathways, there is potential for giving people unnecessary antibiotics. And hopefully my talk will convince you of that. So why is this important? Again, not a surprise to anyone in this audience. In both policy documents in the lay literature, resistant gonorrhea is, a problem. And there's more and more evidence emerging from different jurisdictions that if you look at populations that undergo a lot of intensive STI screening and get a lot of antibiotics, you do tend to see more resistant gonococcal isolates. So there's a field emerging that argues that overtreatment and overscreening may be fueling the problem with gonorrhea resistance. But in the interest of balance, I have to also point out that resistant gonorrhea is also seen in settings without intensive screening and asymptomatic treatment. So although overscreening may be part of the problem, it's certainly not the whole problem. And why is important to talk about it right now? At least in New South Wales, this year we've had issues where we're starting to see extensively drug-resistant gonorrhea isolates, both acquired overseas and locally. So now more than ever, this is an important issue to talk about. So as part of my PhD, in response to this, we asked 3 research questions. The first one is, what are the quantities and indication of antibiotics that are being given in Australian publicly funded sexual health services? Second question is, what proportion of presumptive treatment in symptomatic patient is potentially unnecessary? And what proportion of screening-related treatment in asymptomatic patients is potentially unnecessary? So question 1, so we wanted to look at the trends. We're focusing on ceftriaxone here because it's a kind of a last line treatment for gonorrhea. It has pretty broad spectrum antimicrobial activity. And to answer this question, we use the amazing Access database, which, as we all know, is a really great database that kind of extract data from a network of sentinel surveillance sexual health services in Australia. And for this study, we acknowledged that in Australia, the widespread kind of intensive longitudinal gonorrhea screening recommendations really came, or at least was enforced really well when we started implementing PrEP initially in a clinical study setting until the PBS subsidization in kind of 2018, 2019. So we thought it would be really interesting to study what happens to screening and treatment before and after this period. And so for that, we extracted electronic medical records of the participating public clinics, specifically looking at a cohort of MSM who were attending these clinics for asymptomatic screening. And for this talk, I'll just focus on the cohort level data where we try to stratify reasons for ceftriaxone administration into either ceftriaxone giving for screening diagnoses versus other indications, like if they have symptoms or they were a contact. And for brevity, I will just contract the last 2 categories into non-screening related treatment. So this is the main finding. So if you look from the left to the right of the panel, between 2016 and 2023, the kind of per person year prescription rate of ceftriaxone among these people have almost doubled from 0.1 to 0.2. But what I found really interesting was that not only did the volume increase, the indication for prescription has kind of reversed. Whereas at the very beginning, we were more ceftriaxone was being given for non-screening reasons. By the end of it, we're actually more likely to be giving screening-related ceftriaxone. But the other thing to point out is that despite that, the volume of non-screening ceftriaxone prescription remains high. So in total, we're giving more ceftriaxone. So yeah, we're giving a lot more ceftriaxone, and the indication has kind of flipped. The next question is, what about presumptive treatment? And for that, we did a single site kind of retrospective cross-sectional study where I work, which has been published in STI. And we looked back for around a 2-year period in our center, looking at all MSM who came to us complaining of anogenital symptoms and received presumptive symptomatic treatment. So at the time of treatment, we didn't have any results on them. They haven't had any antibiotics in the last 4 weeks, and we had to do testing on the same day as giving them treatment. And so what did we find? So we found around 1,000 eligible visits fitting that criteria. And I guess the headline items are that we found that 40% of these people that were given treatment ended up not having chlamydia or gonorrhea. Perhaps the part that I found the most interesting is that when we look specifically at ceftriaxone that have been given in these people, around 1/3 of them were given in people who were subsequently negative for gonorrhea everywhere in the body. But not all symptoms were the same. So unnecessary ceftriaxone were actually very unlikely if someone presented with urethral symptoms, whereas if they presented with kind of anorectal symptoms or scrotal symptoms, it's much more likely that they were given ceftriaxone when they didn't have gonorrhea. We followed this up with a multivariate model to look at the factors that were the most significantly associated with being given unnecessary ceftriaxone, and unsurprisingly, anorectal and scrotal symptoms were highly associated, but also other clinical factors that would increase someone's pre-test probability for gonorrhea, such as if they've had gonorrhea recently or their contact of an STI. And this is despite adjusting for kind of other things like other risk factors and whether or not we use microscopy to try to exclude or include the presence of gonorrhea. So what did we find in this study? We found that a lot of ceftriaxone we're giving for covering gonorrhea in symptomatic MSM is unnecessary, more so in, anorectal symptoms. We probably do need better tools for antimicrobial stewardship by increasing the precision of our prescribing. Molecular point of care testing is obviously a promising tool, not a surprise to this audience. And our fabulous colleagues over at Melbourne have done a beautiful RCT that's shown that if molecular point of care tests are available, you will reduce presumptive treatment and overtreatment. But ideally, for clinic flow reasons and health policy reasons, if there's an assay that's rapid and cheap, that would be great. So just whistling through the third question, how screening. Okay, so again, everyone here would know that there has been a very intense academic debate in the last couple of years about the pros and cons and the reasons for doing asymptomatic screening, particularly in MSM. And I won't cover all of that because it's a very complex debate. And also, I'm looking forward to hearing Carolyn's talk, which is following this. I will specifically focus on the issue that some of the STIs that we're finding on screening via nucleic acid testing are actually not viable, which means that they pose no risk of transmission or causing reproductive sequelae, and therefore treatment giving for these infections are unnecessary. So for this, we actually started with chlamydia. The reason for that is because, oh, sorry, the slides are distorted. That's okay. Unlike gonorrhea, we actually already had a small arsenal of tests in research use only that could tell us about chlamydia viability. So for example, you could culture chlamydia, you could design fluorescent antibodies to look at just parts of chlamydia only present when it's replicating. You could look at specific nucleic acids that are only present if chlamydia is replicating. You can look at chlamydia at the membrane integrity, which you would assume is only present if it's viable. And sorry, the slide's cut off. But last but not least, our amazing colleagues over at SpeedX have developed an RNA to DNA ratio kind of real-time PCR assay called Insignia, which Nicole has touched on. Because we already had all these tests, we just thought we should look back at the literature at any studies that have previously reported on a viability in anatomical places that have confirmed chlamydia presence via NAT. And we found 16 studies that were appropriate for the meta-analysis. And I know that this is a talk predominantly about MSMs, but we did stratify all the viability by different anatomical sites. And I guess the take-home message is that actually, if you look at just urogenital sites like urethra, vagina, cervix, actually, the majority of chlamydia that we find that is probably viable. However, if you look at extragenital sites, which are often also places where we do screening in people without symptoms, you do see more non-viable chlamydia. So in the rectum, a third, and throat, a large proportion, although throat chlamydia is quite uncommon. So existing literature supports that actually a sizable proportion of extragenital chlamydia that we find on NAT screening is probably not viable. And one solution may be to implement wider reflex testing of viability that can reduce unnecessary antibiotics through screening programs. So in summary, we're probably giving a lot more antibiotics since the implementation of longitudinal STI care with PrEP, but the rate of so more screening antibiotics, but also a lot of remaining non-screening antibiotics as well. So total quantity is increasing. In symptomatic people, presumptive treatment is leading to a lot of unnecessary antibiotics, especially in the rectum. What's the solution? Is it molecular point of care testing, or is it changing our paradigm to test first and then treat only if something is positive in someone who has symptoms? That's ongoing debate. In people without symptoms, screening probably is giving more antibiotics than needed because there are some infections that are non-viable. some places are more significant than others, and what's the solution? Should we not screen, which in some jurisdiction that's already happened, or should we use technology and introduce reflex viability testing to finesse our prescribing? Again, the debate rages on. So last minute or so, why is this relevant to today? I guess studies like ours really showcases that epidemiological evidence is important because it informs our stakeholders, like everyone in the room today, about how to best finesse screening and testing policies, but also potentially what direction to take assay development into and what are the clinical needs for them. And hopefully we'll create a feedback loop with our collaborators and stakeholders about the clinical and market needs in terms of designing pathways and tests. And for that, I would like to just thank everyone of our collaborators and specifically to my PhD supervisors here at the Kirby. This is a picture of the clinic where I work in, the beautiful Sydney Sexual Health. Thank you.
I was just saying, I think my presentation will be a really nice extension of the two previous presentations. And what I'm going to present today is a repeat of what I presented at the IUCD conference in 2020. And it's really trying to understand, in the context of this debate, should we decrease asymptomatic screening? What are the major points of convergence and divergence on this? How can we get to a bigger common place? And for this crowd in particular, to what extent are AMR considerations even playing into how people are considering the urgency and the importance of doing this? So I think this crowd is pretty conversant with this issue. But basically, we have really high rates of chlamydia and gonorrhea in gay and bisexual men. A lot of these symptoms, a lot of these cases are asymptomatic, as was previously mentioned. And there's a lack of evidence also that these asymptomatic presentations result in clinical harm to cis males, with a good proportion of these infections also clearing, in 6 to 16 weeks. At the same time, there's new evidence, as was shown in those other studies that Arthur put up, that 3x3 screening may be having very little impact actually on prevalence. And so if we're not really getting the benefits that were intended out of this, but there's actually harms, which include, as we've seen, 3x3 screening has really driven up use of antibiotic use. And given the levels of antibiotic resistant gonorrhea, this is quite a large concern. And so one of the options is, should we decrease the frequency of asymptomatic screening? And so in this very contentious field, we really want to know, like, across experts, what are people saying? Can we reach those points of consensus to build on? Can we find out where the divergence is? And then again, find points that we can address those points of divergence. So our three questions were, given concerns about AMR, should guidelines around frequency of asymptomatic screening for chlamydia and gonorrhea and gay and bisexual men of sex with men change? How should we manage asymptomatic cases? And what might be the unintended consequences, and how should we mitigate those? And to answer this question, we use this novel crowdsourcing technology called Polis. It's basically a data collection tool called the Wiki Survey. And so people can enter their own opinions, like short opinion statements, and then other people vote on them in real time. So it's this emerging conversation where people enter what they think is most salient to them, and then people weigh in on whether they also agree or disagree or pass. And so you can engage with this. Any individual could engage with it as little as they wanted or as much as they wanted. So not all statements have the same number of votes. And we used a very extensive snowballing approach to recruit people. We emailed an invite to all members of IUCD, to the CDC centers working on STI, to the WHO collaborating centers working on STIs, to all our Australian networks. And we asked everyone who received the invite, like, please forward this on to other people who would be relevant to participate. So from that, we got 99 people who registered to participate and who actually voted in the exercise. And we had quite a nice spread between the UK and Europe, Australia, which is quite heavily represented actually, and North America. We had majority were women, very high representation of individuals who identify as queer, mainly clinicians and researchers, but this exercise was open to everyone from a professional background working in sexual health. So we did have people working in diagnostics, people working in counseling or community roles, or policy, government policy. And as part of the registration process, we just took a quick snapshot of what kind of characteristics do people have. So our participants self-reported quite a high level of knowledge in terms of gonorrhea and chlamydia treatment, a little bit less so in terms of antimicrobial resistance. And on two opinion statements, while we had 95% of people agree that AMR is a global public health threat, you'll see that only 70% of people thought that AMR in their own country is a threat. So I think we can just keep that in the background of our minds as we see what kind of opinion statements come up. And this is just how to kind of interpret the results. So over the 3 weeks, the participants submitted 91 statements. And for each statement, the software Polis gives you this little figure. So green is the percentage of people who agree, red is disagree, gray is people who pass or are unsure, and white is the percentage of people who just didn't see the comment at all, like they didn't cycle through that comment. And then from all of these 91 statements, I grouped them into categories to see like how can we think about clusters of related statements to see if there's any patterns there. So those are the main themes that I'll be going through. So the very first finding, sorry, the presentation's a bit skewed there, a bit cut off, was the impact of asymptomatic screening on STI prevalence. And here we see absolutely no agreement across the sexual health community, with about half of the participants saying it has not worked to reduce community prevalence, and the other half kind of saying no, like it's key to overcoming prevalence. So like pretty clear split there. And then there were a series of comments about how frequently should we screen. And again, there was no sort of consensus about whether we should move to risk-based screening or encouraging people to screen on demand or offering everyone who wants to screen to be screened in all cases. But there was sort of one point of commonality, and it had some moderate agreement with 70% saying that we could reduce to 6 months. In terms of treating asymptomatic cases, again, there was very little agreement here on whether we should universally treat asymptomatic cases, whether they have clinical significance at all. So the second quote there, chlamydia and gonorrhea in gay men are often without symptoms and pose little or no harm to them. So screening and treatment is not a priority. And you see only have 40% agreeing and 44% disagreeing. Similarly for statements about AMR risk, again, very little agreement. If you look at this quote or this statement, high antibiotic use in gay men has contributed to antibiotic resistant STIs such as gonorrhea. So the less we use, the better. And again, we're only getting about 50% agreement. We're also getting a large degree of uncertainty. So like about 20% of people just pass. And same things for immunity here. The third quote, people seem kind of very uncertain about how to understand what is the role of immunity and natural clearance and whether we need to treat these infections at all. So you've got 36% of people passing on a statement like that one on the bottom. And then the third question was about what are the potential consequences of changing guidelines to screening. And so people were very, very highly concerned and wanted to make sure that even if we stop screening for chlamydia and gonorrhea, that we maintain screening for HIV and syphilis. And this is probably the point of highest agreement in the whole exercise. And then there was moderate agreement about continuing to test and treat or screen and treat for women. Just gonna skip the last one actually. And one of the interesting things around opinions was there was a bit of a convergence about the fact that how much resources is dedicated to 353 screening, and whether we could redirect this more productively elsewhere. So whether or not people sort of thought that it was related to prevalence or anything like this, this was a point of agreement that this is like a very big resource burden. And so this, thinking about going forward, this might be a point where we could kind of rally a little bit around a common goal. And in particular, acknowledging the cost to PrEP programs. And then there was quite a lot of discussion about how community would perceive this change and its acceptability. And I'll go to this on the next slide, but just one little point about whether communities would think this is primarily a cost-cutting exercise. So this is to be tested out more in the community, experts saying no, but and as I was mentioning before, there was like very big concerns in the professional community about how this would be perceived by the gay and bisexual men's community, because they like the reassurance of testing and the safety that it brings to them. And some people were very concerned that this would, that if we changed the screening guidelines, that this would contradict 40 years of what sexual health messaging of what it means to be a good sexual health citizen. But not very many people actually agreed with this statement. So the professional community kind of thinks we can overcome this. At the same time, there was a lot of concern about how you would convey such complex messages in the community. But so really pointing to, and that point of agreement there on the right, that we really need to engage communities on this if changes are gonna take place. And oh, it's fallen off a little bit there. But one of the interesting things about using this POLIS platform is it uses algorithms using, I think, K-component clustering to look at if there are groups of people who vote similarly on sets of statements and differently on other sets of statements. And so with this set of data, they identified two groups. And these are the statements here where there was big differences in how people voted. And so you can see there's sort of group A that falls into the category that over screening doesn't reduce prevalence, it leads to overtreatment and we should reduce it. And then group B is kind of the reverse of that. And then I looked at how the characteristics of people who fell into group A versus group B would differ. And I didn't find any difference except for by geography. So people in Europe were more likely to fall into the let's reduce screening compared to people from North America. This might point to some bigger political and cultural differences, I think, about how we think about like collective risk versus individual risk. I think I've sort of gone through a lot of the implications. So I think I'll wrap up so that we have time maybe just more for discussion. Yeah, and I just did a study on what Australian clinicians think about reducing asymptomatic screening. That's really interesting. So if you want to catch me at any other point on this, be happy to talk about it.
And now we have Matt from ACON, who has kindly joined us to talk about DoxyPEP from a community organization perspective. Thank you, Matt.
I'm hoping I don't spill this water all over the Kirby's nice new computer, but fingers crossed. Yes, great. Good morning, everybody. Hello to the satellite sites. My name is Matthew Vaughan. I'm the Director of HIV and Sexual Health Division at ACON. I have the pleasure of living on Bidjigal land. I grew up on Darug land and I work on Gadigal land, but ACON operates all over lands of the First Nation, Aboriginal and Torres Strait Islander people, and we pay our respects to elders and custodians, past and present, always was, always will be. I must admit, I'm a little bit anxious about giving this presentation today. I would ask that you please hold your pitchforks until the end, but by all means, sharpen them while you wait. If I could also just make a little note that I have got some unpublished data in here that I've stolen from other people, and they've asked me if that I could not share it publicly. So yes, this is not necessarily publicly, but yeah. So gay men are getting a bit of conflicting messages when we talk about STIs. On one hand, they're told that they need to be concerned and be careful. On the other, super strains for gono and syphilis are ending the world, or at least their sex lives. And for a lot of gay men, those two are the same thing. When you think about the history of our community, a lot of gay men actually risked their lives or gave their lives for the sex that they wanted to have, for having sex with other men. Gay and bisexual men don't live in a bubble. The messages that they see impact them. They cause confusion and they don't know who or what to believe. This is why messages from community organisations like ACON and many others in other states and territories are so deeply important. Across the decades, men or gay men and their sex lives have been repeatedly pathologized and often framed as irresponsible, dangerous, or morally suspect, with fear-based health advice as the delivery vehicle. We saw this in the early 80s when gay men were told, look, just cool your heels for a little while, don't have any sex at all, surely it's not worth the risk. We saw this in the early 2010s around the advent of HIV PrEP, that why do you need a pill? You've got condoms, that's perfectly good. And I think we're seeing this a little bit more now with DoxyPEP as well. So a little bit of data. This is from the Gay Periodic Survey, so from 24 and 25. This tells us that there's relatively high awareness of DoxyPEP, particularly in Sydney and Melbourne with more than 60%, but relatively modest uptake in most Australian capital cities. This is a sample that's mostly community attached gay men, gay and bisexual men. So they're very aware of health messages. They're very much what you would consider in the know, or if you use a diffusion of innovation model, they're your innovators or your early adopters. It's important to note that this data is collected at different points throughout a 24-month period, and so there might be some variances. But if we go to the PREPARE study, there's quite high levels of endorsement for DoxyPEP for gay and bisexual men, indicating that our community of innovators and early adopters are actively seeking out ways that they can protect themselves and their partners in a way that works for them, which is actually quite contrary to the narrative that they're reckless or irresponsible. They are aware of AMR and there's genuine concern about it. Fewer than 6% of the respondents indicated that they weren't worried. PREPARE confirms, that's a mouthful, what we've seen in other samples. Gay and bisexual men are already using DoxyPEP. Within the last 2-year period, or within the 2-year period between the two samples, almost a 20 percentage point increase in the people who had ever used DoxyPEP. From the Doxy Aware survey, from the Doxy Aware study, highlighted the urgent need for evidence-based education and resources for clinicians and the community to ensure that it's used safely and effectively. This quote confirms what we know anecdotally, as our health promotion staff are frequently engaging and having conversations with people about DoxyPEP. They're working through misconceptions and misunderstandings. This includes things like how many tablets to take, how many tablets do they need to take and when, the STIs that it prevents, including some questions around HIV prevention, and of course the efficacy. It's during times of deep uncertainty when our communities see conflicting messages and the unknown that they turn to organisations like ACON and many other organisations like ACON for reliable, non-judgmental information. Here's a little bit of the summary of kind of what I've been talking about, but essentially the bottom line is, our communities are often ahead, we've already heard, have really good health-seeking behaviour, and they're often ahead of systems and policy changes. And that creates both a risk for the individual, but also for public health. So if I think about or talk about what we're doing as a community organisation when it comes to messaging about DoxyPEP, I thought I'd just show you a little bit about from our upcoming campaign that will be released later this month through Emanate, funded by the Department of Health and Aged Care, or Disability and Aged Care. The campaign very much focuses on syphilis awareness, with DoxyPEP offered as one of the solutions. You'll notice the campaign messages educate people on the existence and how to use it safely. Information on the referral page provides more detailed information of where to seek out additional support if and when needed. We've given a lot of careful consideration about how to do the messaging, given Australia's strong safe sex culture. We've avoided binary terms like the word safe and instead use words like helps prevent so that we don't overpromise. Of course, this is just one example, and there are already examples out there from Thorn Harbour Health, New Zealand, Burnett Institute, Burnett Foundation, in England and Europe. So we're certainly not the first. I suppose it's interesting, and I often do read or I hear about the concerns about AMR. And it's something that I pay very careful attention to. It's my role to make sure that I can understand what's happening and I can translate that for other people in my community. But I often hear that the real concern seems to come when we talk about sex, particularly sex for gay men. Is it just doctors that are having this conversation about concerns with gay men, or are they doing it with everybody when talking about doxycycline for treatment or prevention? Because again, to go back to the earlier slide, there seems to be a little bit of a pattern here, whereby gay men are framed as irresponsible, dangerous, or a little bit morally suspect, when evidence shows that particularly here in Australia, we are anything but. I would like to, I suppose this is probably just a little thing, is being really careful that we don't prioritize the potential negative consequences over the proven benefits. That's kind of something that I've been working through with the team here at ACON and in a lot of talks that I've given about this. In conclusion, awareness doesn't always equal uptake. So, there is relatively high awareness, but not everybody's using it. We'll continue to refer to condoms and contextualize how and when gay men should consider using them. Gay and bisexual men love options and they like to be involved in their decision-making. And they use this and deploy it in lots of different settings and situations. AMR risk increases with unsupervised usage. Our message promotes how to use it and when to use it safely. I would say that the burden can't all be on gay and bisexual men. We need AMR surveillance, and that needs to play alongside GP education. The benefits of DoxyPEP go far beyond people in our communities. It extends to women and people with a uterus, and this is why we employ a really highly targeted approach. Gay men are responsible and they do very much care. And whatever continues to happen, we will continue to message this to our communities. If we see more, we will update and change our message as we have time and time again. I suppose just to finish up, I would like to say, while I have the privilege and pleasure to present to you today, behind this sits a whole team of work for people working on our campaigns and messages, particularly Chris Williams, who is our project lead for Emanate. But if I could also thank Dr. James McGibbon, Dr. Martin Holt, and Dr. Eric Chow for allowing me to present their unpublished data. Thank you.
Yes. Okay, great. So we don't have a lot of time, but I think for me, one of the key things that stands out from across the board is that there is a question about communicating complexity, I think. I think this I'm going to be doing the discussion, so I'm just going to do my 2 cents and not ask myself any questions. But I think in a context where we teach science as if it's a silver bullet and we don't expect teaching people to sit with uncertainty is really problematic. Like, the world's uncertain. That's just the way it is. And the world is constantly becoming and moving. And I think, I guess, these ideas about certainty and that we can kind of control the world and mastery over the world informs a lot of the things we talk about and the way we see things. So I guess taking what I'm saying as being true, sorry, I'm not giving you an option, taking that idea of science and the world as, to borrow from STS scholars, as always becoming, how do you think we should communicate health messaging and sexual health signs in such a context, I guess, is, I think, the big question that we're thinking through. So Matt, did you want to go first?
Yeah, I think particularly for gay and bisexual men, they're used to dealing with this complexity, and this has been in their sex lives now for the better part of 40 years, possibly even longer. I think it's helpful to provide, as we always do, we know, health information that's clear, easy to understand, and that allows them to walk them through the choices that they have and allow them to make their informed decision is always helpful. I think one of the things that we've really used in our campaigns is to provide a relatability factor. So we often use real talent and real people in our campaigns to talk so that people can see other people using it and how they deploy it in their life to help give that perspective of things that they haven't necessarily thought about. A lot of people don't know necessarily what they're after until they're shown what a possibility is. And when they see that possibility, they're excited by it. So I think that absolutely has a benefit, but those are just some of the top ones that come to mind.
Because it's interesting, like even before this presentation, when I've been doing field up for this project, people ask me for certainty, and I've just gone, I don't know what's going to happen with DoxyPEP. I can't tell you what the future outcomes are for sure. I can tell you watch this space, but I cannot give you a certain answer about it. And so it's been really interesting, right, for me having to tell people I don't know with certainty. Yeah.
This also happened with HIV PrEP. So we didn't know exactly what was going to happen when we implemented it in New South Wales or in other jurisdictions around the world. But what we said to people is that we would communicate through it. So we did a lot of education forums, there was campaigns, there was GP education. And as the data changed and as our confidence grew, so did our language. We went from referring to it as kind of 97% effective to talking about it as safe. That was a really big thing in Australia that just provided people confidence that this is a form of safe sex. That's a form of hesitation that we are being really careful here with this DoxyPEP. We don't use that word lightly.
The other, I guess, Arthur, for you, I guess reducing testing, right? Like kind of in like DoxyPEP, what do you think about messaging around that, and what do you see as limitations and also possibilities for that messaging with communities?
Oh, I'll stick to my patch, which is the implementing clinical side of things. In the last conference in Montreal, there was a really great session from some of the European settings that have already reduced screening, and all about how they prepared themselves, the frontline clinicians in conversations with patients. So they deployed lots of resources, videos, mock interactions, and to your point about how to navigate uncertainty with patients. So I think if we in Australia, if we are to go down this road, I think there's a lot of prep work that's needed, and like preparing people like me and people I work with in the clinic about how to have these conversations so that we have a united message.
I mean, it's interesting because I know when I've spoken to people, this often doesn't get included as a concern for MSM. But I started asking people, I was like, are you concerned about reproduction and what might happen if you've got an infection? Because we often imagine queer people as being not like we don't factor them making kids, right? And so I think that was something that some people were like, oh, I don't care about it. But a couple of people said, actually, that would matter to me. And what is going to happen if an infection is in my body? So, yeah, I think that's an interesting thing to think about, that the messaging also needs to speak to those concerns. And we shouldn't not see that as something queer people want to do. I was going to say, Terrellyn, did you want to speak to the same question about how we go about messaging a potential reduction in testing?
Sure. Can you hear me? Can you hear me?
Yes, but maybe a bit louder.
Okay, I'm going to yell at this room.
Yeah, just go for it.
This isn't really a solution, but it's a larger problem that I think that we all need to grapple with public health messaging. And I think it's this idea around how do you grapple with risk that's on a population level versus an individual level. So drawing from the interviews that I'm doing with clinicians is when they have a patient right in front of you, and this speaks to Matthew's thing, the benefits are so clear for that patient, but the consequences to the public are uncertain. And it's the same with climate change, right? It's really good for me to buy a cheap car and I will choose that car. But when we all do that, and I think to me, this is the complexity that we need to learn how to communicate globally on everything, but including in the STIs with regards to antibiotic resistance. It's not just to communities, it's also to clinicians, it's to us, because we know that vague risk doesn't affect our behavior. And so we need to figure out, the people who work in AMR advocacy, how to affect behavior and attitudes around risks that may or may not apply to you individually, and then allow for space for people to make individual decisions, but have that bigger framework.
Thank you very much. We technically are at time, but there are a couple of questions at the back. So if we're happy to go 5 minutes over, is that okay, Tanya? Yeah.
Absolutely. If you
Hi, it's John Caldwell from the Kirby Institute. Lots of really interesting presentations. Just maybe a bit getting away from, I mean, not getting away from, but in addition to the community and communication issues, this is more, I guess, a clinical question, and maybe Arthur and others have some thoughts about the idea of the different options of more reducing screening or taking a different approach to testing before treating. Is there a consideration for doing it differently at different sites? For example, you might think about, you showed the data that with urethral discharge, it was more often appropriate, and that's where you have the concern about the resistance taking place, whereas resistance being caused, but with rectal, it's much harder to do that testing because it's harder to get the testable specimen. So you might even though you're doing a lot of over treatment, it's actually maybe more practical to keep up with screening there, and it further complicates the message for people to understand that. But is that something that's being considered?
Yes, I agree with you, and I think we didn't have time to, but I will in other talks I elaborate on this point, which is I think the urethral discharge story is quite interesting because it's a clear example of how tests don't perform the same in different places. Because like we have point of care tests, it's called a microscope, right? And it's actually really accurate in the urethra, which is why it's not a problem in the urethra. It's not very accurate in the throat and the rectum. So I 100% agree with you. I think whatever test we have, the implementation has to be considered differently in different anatomical sites of population. But then circling back, and the communication becomes more difficult. Like if you have a uterus, but not a rectum, or you have a rectum, but which part of you use for sex, and the messaging might change. But I think the communication is hard enough as it is, that I think it's a big piece of work to get it right. Yeah.
Hear me? Yeah. I'll be very quick actually. So Rebecca Guy from the Kirby. And Alexis, I'm going to put you on the spot. Just so I'm aware that Asham is organising the screening consultation. So it was just great to listen to all these talks and thinking how they could contribute to that consultation. I'm just wondering, Alexis, if you wouldn't mind commenting.
Thanks, Beck. Yeah, so end of the year, we do have the testing screening workshop roundtable. I think some of you are already involved, and definitely from this, it's been really exciting to see. I'm glad I came, just to make sure some of these are included and people included. Also, maybe just to note, the DoxyPEP decision-making tool was launched in March, probably ahead of the guidelines. The fuller guidelines only at the end of this year, maybe the beginning of next year, probably. But already the uptake, without any comms, the uptake is quite high. I saw some numbers earlier today. At least 1,000 users who regularly visit the guidelines, which is also, I mean, the decision-making tool, which is interesting.
Kat, did you want to speak?
Yeah, just a couple of thanks for the great presentations. And yeah, like John, Arthur's talk made me really think about sites. And I think, clearly testing for chlamydia at the pharynx is something, or screening is something that we could fairly easily get rid of. There's little benefit, there's little evidence of viability, and it seems there that's an easy low-hanging fruit. I just wanted to make a comment about DoxyPEP because it obviously is an emotional topic. I don't think it should be. I think it should be just very practically addressed. Unfortunately, it's a simple mathematical formula that antibiotic consumption is directly related to AMR. So we don't see AMR in communities where we don't have access to antibiotics. So that's irrefutable. And unfortunately, I think HIV PrEP is not a good example because we were really able to measure potential harm there. We had one target organism. We didn't have a lot of target organisms. You could track development of resistance in HIV, whereas we've got huge numbers of microbes in our communities that we can't necessarily capture the impact of widespread doxycycline use on as easily. But I think, the bottom line is that we really just need to understand and accept that DoxyPEP will do harm, whether we are able to measure all of it or not. But it's really a case of whether the benefits outweigh those harms. And I think we need to reframe that conversation from, denying that it will do anything and accepting scientific uncertainty. And there's without a doubt there will be people who will benefit from it, and it will be a case of us seeing, how much benefit our communities derive. That was really, I think, all I wanted to say about that.
Actually, to follow up from what Kat said, not just the resistance. If someone is quite a regular user of the DoxyPEP, it's affecting their biome. So what are the long-term consequences of being on DoxyPEP for 2 years, say, if you're just very active and want to take that preventative measure? So, are there public health measures in place to monitor people's biomes or their health, and what's the consequence of taking that long-term?
Thank you for that. We have a lot of work in that space, I imagine.
Yeah, great. So thank you very much to our speakers.
Okay, so I might start the next session up. Hi everyone, I'm Shivani from the Doherty Institute here in Melbourne. I'm really excited to start off this session. Louise is going to share her slides for me. This is a bit of a different session because we're talking about the learnings from the Point of Care 2025 conference in Thailand. And I'm really excited about the session because I didn't get to go and I was very jealous. Perfect, thank you. Yes, so such a huge success. Next slide, please. Just to summarize the efforts and the success, there were 150 abstracts submitted, 44 oral presentations, 58 posters, and 3 days of wonderful research. And I think why we wanted to talk about it today is part of the hub. The hub really facilitated some of our EMCRs being able to go. And so today we're going to hear from 3 of those recipients of this award. So first up is Louise from the Kirby. Go for it.
Oh, thank you. I'd just like to start by acknowledging the Bidjigal people, the traditional custodians of the land from where I'm presenting from today, and pay my respects to elders, both past and present. So my name is Louise Chow. I'm in the SERP team at the Kirby Institute, and I received a scholarship from the AMR Hub to attend the POC 25 conference in Bangkok. And I just want to share a few insights from the conference. I'm a molecular biologist background, so I was really interested to go along to the conference and see some of the tests in the point of care testing space. So there were 2 presentations that gave a really good overview of the point of care testing space, Harisha from FIND and Tanya from the Kirby, and that was excellent. This is just one slide from each of their presentations, and you can see that there's a lot of point of care tests, and there are multiple slides full of really cool new point of care tests. But there were also some presentations that did highlight gaps in the point of care testing space, and these gaps could have been point of care tests that didn't perform as desired or diseases for which there are no point of care tests available. And I think sometimes when there's gaps, it can become a bit finger pointy, and we can fall into pointing fingers at each other and assigning a bit of blame. But I think that's why the point of care conference, and also why this AMR Hub is so important, because it really brings together industry and researchers and end users to work collaboratively and bring really effective and considered tests to the people who need them. But what do these tests look like? And throughout the conference, there were some themes that kept coming up of what the ideal point of care test would have. And I just wanted to touch on a few of those today with some examples from the conference. So the first feature that came up was portability, and this is really vital if trying to reach remote communities that don't have the resources for a permanent point of care facility. But if you consider that traditional pathology is performed in a highly controlled laboratory with expensive, delicate equipment, translating that into something that's portable is quite a significant task. And there's 2 presentations that highlighted the importance of having a portable point of care test. The first was this one by Dr. Samantha Tinze, who's working on tuberculosis diagnostics in the Philippines. And I loved this presentation. She has so many great photos up on her website, which I've included below. But they actually transport the point of care tests island to island using boats. And they use a ultra portable chest X-ray for TB screening, and then they confirm with the MolBio TrueNat, which is a portable PCR-based machine that can be battery or solar powered, room temp stable reagents, sample to answer in an hour. And it's also able to determine drug resistance. And using this approach, they've managed to go from testing 200 patients per year prior to 2022 to 6,000 in 2024. And they can provide on-the-spot treatment for TB that's susceptible to drugs, and they can provide a referral for treatment for TB that is drug resistant. The second presentation I wanted to touch on was by Dr. Paulus Ribra. He presented on the HPV test and treat work that they're doing in PNG, and they've fitted out basically a mobile lab on wheels. So that picture is their outreach truck, and it has GenExpert PCR machines in it, and it has a bathroom for self-collected samples, and also the treatment testing takes about 1 hour, and treatment can be administered in the same day. So I think these examples just really highlight where being able to take the point of care test to the people that need the testing is vital in reaching those communities. The next feature of the ideal point of care test is that it should be fast. And the first presentation I just wanted to touch on was by Tristan Clark, who presented the work from the FluPoc trial, which was a trial using the BioFire film array, which has insane multiplexing of 22 targets in 45 minutes. And they tested patients presenting to the hospital, presenting with respiratory symptoms. And unsurprisingly, patients who were tested with the point of care machine received their results much faster, within about an hour, compared to standard laboratory testing, which was about 24 hours. And because of that, patients who were tested with the BioFire film array received the correct treatment sooner. They were effectively isolated, and they had improved clinical outcomes, including being less likely to go into the ICU. And I'd also just point out, it wasn't included in Tristan's talk, but BioFire has now an even faster platform, the Spotfire, which produces results in 15 minutes. So we can definitely see this trend towards point of care testing becoming faster. And the second presentation was Dr. Jabel Foster, who presented on work up in the Torres Strait at the international border with PNG. This was such an engaging presentation, and she really stepped us through the problems that they encounter there when trying to access healthcare. People from PNG who cross the border for tuberculosis testing cannot be recalled for results and treatment. And this means that if patients don't receive their results and they cross back over the border, they can be lost to follow-up. They can be very difficult to locate for results and treatment. And here they implemented the Cepheid GenExpert, which was able to also diagnose TB and resistance. And it really transformed tuberculosis diagnosis in the Torres Strait and brought testing to them, and they were able to provide same-day referrals and treatment decisions. The next feature I'd like to touch on is sample type. And sample type is so important because it affects performance, acceptability, and ease of use. And the validation and registration of each sample type is a lengthy and costly endeavour, so it really needs to be considered carefully. Throughout the conference, it was clear that the ideal point of care test should allow for self-sampling or self-testing, and I'll give a few examples. The first being was this presentation by Brittany Choate from Saliva Direct. Saliva Direct's a not-for-profit that spun out of Yale, and they focus on developing tests with saliva as the sample type. And they chose saliva because they found it was a non-invasive sample type that can be self-collected, that nucleic extraction is not always needed, which simplifies workflows. It's cost-effective, it's stable, and it's very versatile, and and it can be used for many different pathogens. Following COVID, they did a survey of 125 countries and asked them what their preferred sample type was. And you can see that there definitely is a global trend that saliva is the preferred sample type. And they now use this approach mainly for COVID testing in a range of formats, including the mobile lab in a van. And they also use these vending machines, which allow for anonymous 24/7 collection of testing kits. And patients can then self-collect their saliva sample and return it into the vending machine where it's taken for testing, and then they receive the results via the method of their choosing. And they found that this approach is very useful for reaching rural communities. And I had a chance to talk to Brittany at the conference, and it seems like Saliva Direct is very keen for collaborators, and she even said she likes writing grants. So I've included their website on the bottom of this slide if you'd like to have a look. This came up quite a few times throughout the conference, but I'll just touch on the two presentations by Raina and Natea, who really highlighted the importance of self-sampling and self-testing for transgender people who may be reluctant to have health professional collected samples. And they both really championed community-led programs and shared insightful stories of patients being reluctant to accept testing until it was in a format appropriate for them, showing that here self-sampling is essential in providing diagnostics to these groups. And lastly, I'll just touch on this self-testing device by Atomo, which has a built-in Lancet and integrated buffer delivery system. And this is really good because people generally hate finger pricks, and so making it as streamlined as possible is ideal. And there were 3 presentations that shared programs using the Atomo self-testing device. Elsie Williams from the Burnett shared the new in development syphilis self-test, and Bo Newman and David Lewis presented on programs that utilize a HIV self-test, which is distributed via post or via vending machines. Reducing barriers to access for these tests is vital and only possible through the self-testing format. And I only had time to touch on a few features of the ideal point of care test today: portable, fast, self-sampling or self-testing. And I also discussed a bit about the tests that were able to determine drug resistance and how important that was to administer correct treatment. But there are many more features that really need to be considered, including affordable, easy to use, satisfactory performance, which is normally benchmarked against laboratory performance, and open source. I really enjoyed in one of the industry sponsored sessions, someone asked if they would make their platform open source, to which they very politely said no. But really an open source point of care platform would transform diagnostics in resource constrained settings and for diseases which have no or few available diagnostics. And this seems like a big list, and it's clear there's still a lot more to do. But I wanted to end on a lighter note, because I think sometimes when looking at how much more there is to do, we forget how far we've come. And looking at CT and NG as an example, we can see how testing has evolved over the years, up until 2023 with the VISBE medical sexual health test, which is an instrument-free home test. And I just think this is a really exciting space to be working in, and I'm looking forward to the future. And big thank you to the AMR Hub and Rapid for the scholarship to attend the conference. And I'm also just going to spruik. We have a STI point of care landscape review that we're working on. So AMR Hub, Kirby, and Find, and many other global partners. And so if you are developing or marketing a point of care test for a sexually transmitted infection, you may be interested to put your information into the survey so you can be included into it and included into a peer-reviewed report. Details are on the AMR Hub website. Thank you.
Next speaker is Jack Lynn Prestige, joining us from the Doherty Institute. Thank you. Thank you, Shiv. And again, thank you to the Hub for funding my trip to Thailand to go to this conference.
We've been asked to present some learnings, so I'm going to share more about multiplex testing and wish list to workflow. Louise has already highlighted some of those examples that Tanya provided one of the plenary sessions. I've stolen her slides, but just to demonstrate the range of multiplex point of care tests that are out there. So when we're looking at multiplex testing, I guess it's the detection of multiple analytes, often pathogens, but also could be different markers, for example, for antimicrobial resistance, in a single test. The benefit here being, rapid and comprehensive diagnostics, using one sample being the ideal what you want. And so you could detect co-infections, but also differentiate similar symptoms. For example, respiratory symptoms that all may be the same. How do you tell what the causative pathogen is? And there were so many different platforms out there on the market, either in the research stage or fully implemented at different points in time. And we saw lots of presentations on this and the different availability of them. And why I wanted to focus on this is because it was shown, I know this is a big table, the details aren't super important, I'm not rolling through all of these, but there were so many examples of point of care testing programs presented at the conference that were really trying to integrate into a service, either an existing service for something else, like antenatal care, and adding testing in there. But to deliver these, they needed so many point of care tests. That included multiplex tests, that also included singleplex tests. And to scale these, you need, and to deliver a comprehensive service, it requires a lot of different tests. That means different training, different quality management, extra cost of having multiple different tests to do multiple different purposes. And so this really led me to think about how we need more multiplex tests or different multiplex tests to really wrap around the services required. So I'll just point out a few examples here. Louise has already touched on the Thailand experience of key population-led services. Having STIs, HIV and hepatitis C integrated meant that there's a massive range of point of care tests required to do that. Similarly, there were presentations on triple elimination, so the elimination of HIV, syphilis and hepatitis B virus, particularly in antenatal care settings from East Timor, but also the Menjaga studies. There were a few presentations on this, which is in Indonesia, looking at the different HIV and syphilis tests required. But Indonesia in particularly, there was a poster about this that talked about the supply of the test being really hard to get in countries as well. So some clinics could have the HIV and syphilis combination lateral flow, but they also had to have the single plex lateral flows available because they couldn't always have the supply that they needed. And so we need options is kind of the big thing here. The example from the First Nations molecular point of care testing program, aware there's a whole bunch of people at the Kirby that work on this and it's not me. So apologies if I'm misinterpreting things. But a lot of the Aboriginal and Torres Strait Islander community clinics that have these gene expert platforms have now integrated having both STIs and respiratory infections. I think it's about 70% of clinics now offering both. Most of them are all on the gene expert, but there's still singleplex for trike, for example. So we're not at full combination of all these different things. And so I just want to highlight that there's still a long way to go in if we want to develop multiplex tests to really allow for one test, one specimen to give a one-stop shop for a patient. And so I'm going to summarise across all of those presentations to say that what we heard from healthcare workers was they want cheaper, easier, accurate, fast, reliable diagnostics. And Louise touched on this. But the clients, they also want fast results. They don't want lots of finger pricks or lots of swabs. They want it to be as simple as possible. And they want a one-stop shop, not a we can test for this, but we're missing this thing. You have to get a lab test for that anyways, because that doesn't actually save them time or convenience of going to a clinic. And so there are ongoing challenges in this space. So obviously your financing, when you require so many different point of care test kits, that costs a lot of money, and there's extra costs related to outreach programs as well. The workforce has to be trained and trusted to deliver those services and act on those results, and we have additional complexity with that. But then also integration, so who has the responsibility for the testing and the result reporting? And then at the systems level, and Chai, the Clinton Health Access Initiative, spent a lot of time on this in terms of talking about how you maximise the value of the platforms or the instruments to deliver lots of different types of testing on those. And then the regulation to deliver this. There were lots of talks about how peer-led services are really, really crucial for engagement with clients, but the regulation is needed to allow this non-medical workforce to deliver these tests, to potentially diagnose, connect to medical professionals for treatment, and some even were pushing for peer-supported self-testing to kind of eliminate those barriers. So my learning was that design point of care tests with stakeholder input from the start to ensure that these are user-centric and workflow driven, and they're the point of care tests that get to market. But the big challenge here is that the wish lists are not universal. And you saw in that table that often they were all doing HIV testing, there were lots of syphilis, but they were paired in different ways. So every country and healthcare setting has different priorities for their diagnostics, and the most relevant pathogens really depend on the local needs, the epidemiology, and the health system capacity. It's perfectly valid to have HIV combined with hepatitis B or hepatitis C or syphilis or on its own. They're all needed in different contexts. And so how do you design one multiplex test that will fit all of them? And should you? And I guess the answer is that including irrelevant targets leads to confusion. We've seen multiplex tests that have, tens of targets, up to 22 in the BioFi that we saw. What about if you don't have the ability to act on those results? You don't have the knowledge to do something about them, you can't treat them. Is that helpful or confusing? And multiplexing itself technologically can reduce the performance of some targets. So the targets that we really, really care about with the highest consequence that we want to treat, we're reducing their performance by compromising with all these other targets that we may not be able to do something about. So because the test performance also depends on the local disease prevalence, so for example, a low prevalence setting, the chance of a false positive increase. And so in a low prevalence setting, let's say in Australia of hepatitis B virus, if we were to implement the same kind of triple elimination multiplex tests as in some countries where there's a higher prevalence, we're more likely to have false positives in Australia that could cause unnecessary stress for potential diagnosis that is then not confirmed by laboratory testing. And what harm does that do as well? So my learning is there's no one size fits all. And while we ask for multiplex tests, the selection of targets is going to be really challenging because it needs to match local guidelines, workforce expertise, the supply chain and the cost realities, and the disease priorities, which are all things that are very, very context dependent. And so I think there are some opportunities that were presented to this. So there was another plenary by Dr. Polman Chan from the WHO, and I've stolen this slide from her as well, that talked about some new guidelines coming out from the WHO next year that are looking at multiplex testing and multi-disease elimination. The reason for this, because countries and regions are requesting guidance for multiplex testing, especially around the triple elimination. But these are already being used in many places. They're being developed by industry, but there hasn't been kind of a global framework for this yet. And there's been a lot of positive experiences, particularly with getting more syphilis testing happening with the combination of HIV and syphilis dual tests, especially in antenatal care settings. And there's emerging evidence with other multiplex tests, and that we could really consider how much benefit this would give. This was after the conference, but just to note that the WHO pre-qualified the first triple diagnostic test for HIV, hepatitis B and syphilis towards that triple elimination goal. But again, this won't be relevant in every setting depending on what the different pathogens of interest may be. But this company also has them all in singleplex in combination. So how many times are you going through that regulatory process? And are all companies able to do this? And is this realistic to ask in our industry? So I think the learning here for me was that global policy is really helpful to guide the test development of multiplex point of care tests, and it supports the market readiness. We need global funders that are willing to pay for these tests. We need local governments to pay for the tests, to be ready for their workforce to deliver them, to have the data and systems to implement them and record those results. And as we introduce more multiplex testing, potentially without guidelines for those pathogens, how do we go about managing these things? And so we need those come together with the policy and the practice. So now to summarise everything all together, I think multiplex testing design, there's technological challenges. We're going to have trade-offs with sensitivity and specificity with the more targets you add. There's more potential for cross-reactivity. And the sample and reagents, it will depend for different sample types. Sometimes you need blood, sometimes you need a swab, or they'll work better for one or other. How do you multiplex when you know there are sample limitations? And so the learning really here is that regulatory clarity is going to be really important to understand how many times companies are going to have to go through this process and what the key differences are. They're not all necessarily the same. In implementation, to integrate this into clinical pathways and act on results is really important. And so that will need staff training, that will need a change of scope of practice, potentially peer workers that are doing this that are really community-led and have a lot of experience in a specific area, how that gets broadened out and how we keep training. And the data management and reporting of results will be critical. So we need robust evidence and standards for implementation in different settings to really help support the implementation. But to make it adopt at scale, we're going to need really value proposition for decision makers, multiplex testing is more expensive, or it's getting cheaper and cheaper, but it also causes more hurdles for industry in terms of regulation, and it causes more change to workflow. You've got more things you need to manage and treat in a clinic. So that makes it a little bit more complex. And so we're going to have to look at the cost effectiveness and the benefit to having disease agnostic platforms like the Gene Expert, where you can have lots of different kits and menus, and that's really important. But there's going to be a tension between integrating all of different testing and protecting funding for specific diseases and our priority diseases like HIV, malaria, tuberculosis. The more we kind of dilute that, is that a good thing or a bad thing? And there was some active discussion on that at the conference. And so my learning there is that we're going to need a lot of early health technology assessments of multiplex tests, and that will help us drive adoption of these when they are cost effective and useful. I'm finishing there, but just my big take home conclusions is that the landscape is really changing and it's advancing towards multiplex testing. This is happening more and more, but there are key operational, clinical and systematic challenges that we need to address before we are to adopt widely. And to realise the full potential, we really need to think about this because there's great benefits to community and to clinics for having integrated testing that can offer a one-stop shop for services. This is a photo from a session that I presented some of my PhD work in, so again, wanted to thank the organisers and everyone for the opportunity to go.
Different regulation here in Australia. What was your feeling about what could and couldn't be done in Southeast Asia relative to what we can do in our research space?
Yeah, I think there's a lot to that, I guess, in terms of there's no global standard for some of these regulations. And so this same point of care test is going through each regulator. A big factor is getting WHO pre-qualification for them to be used in low middle income countries that don't have their own regulators and rely on that WHO pre-Q for funding and to implement it within there. And I think it's challenging to work out the market size. We often hear in Australia tests don't make it here, even when they're really good, because there's not enough of a demand for them. And so I think there's challenges. Unless we have a single worldwide, only there's one regulator or everyone acknowledges and reciprocates each other's regulations, then that would really help. But we have different things in different countries that look at everything from how your instructions are written and how the language that's used, which does require jurisdictional differences across countries. And so what is allowed to be tweaked and what is not allowed to be tweaked without requiring a full review. I think it's a whole system upheaval and change that would be required to do so. But I've never gone through regulatory submission, but I think for a lot of the industries that they speak about and the companies, that is a massive hurdle because it's a lot of cost to go through that regulation process and to duplicate that for different tests. And I think we saw that in the COVID example in Australia, that point of care tests that was allowed first for COVID and then the self-test for the antigen tests. Often they were the exact same technology. They had to go through the TGA's process and the ARTG with a different instruction for use to become a self-test version, but they were essentially identical. But that's an extra test being listed and an extra cost and all of those kind of things. Sure. And it is important because it's different people using it, but that's just a lot to think about.
Yeah. Brilliant.
I will hand over to Rabia and Andre, who are co-chairing this session. Can you just see the slides? I just want to check first.
Yes, I can see the slide.
Okay, thank you. I believe Qinglu's sharing remotely.
Yes, Tanya. Sorry I couldn't join you on site today. I have a bit cold and yeah.
No problems.
No worries. All right, hello everybody. Welcome back after lunch. I hope we're all rested and recovered. Now we're on to session 4, developing a business case and generating the evidence. I personally believe it's very important to make sure we have data-driven decisions, and here we have a lot of people to talk about those. And I'd like it for Qinglu Cheng to take it away, please.
Thank you, Andrew. Can you see my slides now?
Yes, I can see that, Qinglu.
Great, thanks, Bia. So good afternoon, everyone. My name is Qinglu. I'm a research fellow founded by AMR Hub and Stride CRE at the Kirby Institute. And today I want to talk about using economic evidence to inform commercialization with an example on the cost of unit study of Mycoplasma genitalium. Before I start, I would also like to acknowledge the Gamilaratical people of the owners of the traditional land where I'm at today, and pay my respect to the elders past and present and emerging. And I'd like to begin by discussing why the industry needs health economics evidence. The commercialization procedure of a product can be generally categorized into 4 phases. In phase 1, the economic evidence can be used to understand current market. For example, cost of unit study is usually conducted to understand who are the target patients, what is the prevalence of the disease of interest, and what healthcare resources are usually used to manage the diseases. This type of information is vital for us to understand whether there's an unmet need that a new product could fit in. In the second phase, the economic evaluation studies can project the future market. So with economic evaluation studies, we'll be able to investigate whether additional investment in new products such as diagnostic tests can be offset or even outweighed by future cost savings from better health outcomes such as reduced AMR or improved patient outcomes. Usually we would plot this cost effectiveness plan. So anything that lies on the left-hand side of the willingness to pay threshold would be deemed not cost-effective and not worth funding. If the product lies on the right-hand side of the willingness to pay threshold, then we'd say this product is cost-effective or can be even cost-saving. So this information can inform the go or no-go decision usually involved in the early phases of product development. The economic evidence can also be used to support regulatory submission. And for example, in Australia, the Medical Service Advisory Committee usually would collect all relevant information on safety, clinical effectiveness, cost effectiveness, and total cost. And before a medical service or diagnostic test can be listed on the Medicare benefit schedule. In recent years, there's a growing emphasis on equity considerations in decision making. For example, in the UK, the National Institute for Health and Care Excellence is trialing to include equity evidence in their guidelines for smoking cessation. Similarly, Canada has recently launched a new strategic plan that included equity as one of the guiding principles. So with both effective and with both efficiency and equity evidence, we will be able to tell whether the new product could lead to a win-win situation where it's both cost effective and improves equity, or the product could be just cost effective but harms equity, or it could be either loses or lose-win situation. So even when a product enters the market, we still need economic evidence to maintain market share. Over time, with new treatment pathways, competitor products, new registry data or claims data become available, the economic models previously developed can be continuously updated to inform whether a new product is needed to replace the old ones. Next, I will just talk about the cost of illness study we recently completed for Mycoplasma genitalium. So Mycoplasma genitalium is a bacterial sexually transmitted infection. It is often asymptomatic and is not a notifiable condition in Australia. And due to this feature, the incidence and prevalence data on Mycoplasma genitalium usually derived from a small-scale observational study or modeling studies. Mycoplasma genitalium has been found to be associated with urethritis in men and pelvic inflammatory disease, PID, in women. According to a recently published systematic review and meta-analysis, Mycoplasma genitalium was detected in 1 of 10 women diagnosed with PID. The presence of Mycoplasma genitalium has also led to a 67% increase in the odds of PID. Currently, the national and international clinical guidelines for managing Mycoplasma genitalium does not recommend asymptomatic screening. This is mainly because the natural history of Mycoplasma genitalium is not well understood. Moreover, there is an increasing risk of antimicrobial resistance in Mycoplasma genitalium. And at the moment, a single dose of the macrolide azithromycin has been widely used as first-line treatment for Mycoplasma genitalium. However, the rapid increase in the macrolide resistance in Mycoplasma genitalium has been observed across the globe. In Australia, for example, there has been a sharp increase in the prevalence of macrolide resistance from 19% in 2010 to 57% in 2024. And at the same time, the resistance to fluoroquinolone is also rising from 10% to 35%. So this rapid rise in antimicrobial resistance is affecting all available therapies, leading to clinicians encountering an increasing number of patients with untreatable infections. So one resistant guided therapy might provide a way to address this issue. So resistant guided therapy uses resistance assays and a diagnosis to guide the selection of effective first-line antibiotic treatment. Studies have found that the resistant guided therapy has the potential to reduce the selection of macrolide resistance, and our colleagues have also demonstrated the cost-effectiveness of resistant guided therapy among women and men who have sex with men. Well, our study, the cost of illness study, aimed to quantify the economic impact of managing Mycoplasma genitalium with alternative treatment strategies, which is the resistant guided therapy versus non-resistant guided therapy. Our cost of illness study adopted an incidence-based approach, so we captured the cost from diagnosis until recovery. And we focus on 3 subgroups: the non-pregnant women, men who have sex with men, and men who have sex with women. And our study adopted a healthcare system costing perspective. So we only included the cost of testing, medical consultation, medication, and hospital admissions for any Mycoplasma genitalium related complications. The time frame was set to 12 months, and we assume that, once cured, there will be no infection recurrence. We rely on a range of data sources, including published literature, pharmacy price, national hospital cost data collection, Medicare benefit schedule, and the pharmaceutical benefit schedule. This is the decision tree model we use to reflect the clinical pathway of managing Mycoplasma genitalium under resistant guided therapy versus non-resistant guided therapy. And here are the results from our baseline analysis. So if you look at this column, which shows the expected, the average cost of managing a patient, you'll find that the resistant guided therapy cost about the similar amount to the non-resistant guided therapy. It is slightly more expensive to the resistant guided therapy is slightly more expensive for women and men who have sex with women, but for men who have sex with men, the resistant guided therapy is more cost-saving compared to non-resistant guided therapy. We also conducted one-way sensitivity analysis to assess which variables have the greatest impact on the cost outcomes. So this graph shows the sensitivity analysis results for women. And as you can see, the cost of Mycoplasma genitalium related complications has the greatest impact on the cost outcomes. The higher the cost of the complications, the more likely that the resistant guided therapy would become cost effective. Other variables that have a great impact on the cost outcome include the cure rate of first-line treatment under non-resistant guided therapy and the cost of first-line resistant guided therapy. So our study found that the average cost per person managed by resistant guided therapy are comparable to those of non-resistant guided therapy. The resistant guided therapy would become more cost-saving when the cure rate of first-line non-resistant guided therapy declines, which is the case in the real-world setting. The resistant guided therapy would also become cost-saving when the cost of first-line resistant guided therapy are reduced. This is because the moxifloxacin, the medication used to treat macrolide resistant infections, are currently not subsidized through PBS. The patients can still purchase the medication at the pharmacies, but they need to pay a price higher than it would be if it's listed on the pharmaceutical benefit schedule. So this finding underscores the importance of generic formulations and the importance of revisiting the pricing and the subsidization policies for effective antimicrobial therapies in response to evolving resistant pattern. So to conclude, the resistant guided therapy has the potential to slow the spread of macrolide resistance without imposing significant additional cost. And our study findings can support future cost effectiveness and or cost benefit analysis of new approaches to managing Mycoplasma genitalium infections. And that's all for my presentation today. Thank you for listening.
Thank you so much, Xinglu, for the presentations.
Okay, anyway, so let's start take two. So today I'm going to talk about modeling. In particular, modeling the impact of viability testing in reducing antibiotic usage for treatment of STIs. And the joke was, lunch up. I always got like presentation after lunch, and it's really, really hard to keep focused. But that's how it goes. So, as I said, before I dive into the result, I would just like to have a few quick slides into the concept of modeling. Now, by definition, modeling is, well, a model is a simplified representation of reality to help to describe the causal relationship between a mechanism. And there's many different types of modeling. So there's economic modeling that Chinlu just showed earlier, and there's also climate modeling, other financial modeling. But what I am interested in is the epidemiological modeling, which integrate the behavior, biology, and clinical data to describe the transmission of certain disease through a population. So for example, we have a diagram here, which is, you can say, is a sort of model for a certain outbreak. Now, I guess, let's jump. Anyway, so I guess we got this modeling tool available. So I would just like to quickly introduce why we use modeling in the first place. Now, typically, modeling can be used for three purposes. So some modeling can be used to understand the current event or past event, such as testing the hypothesis. For example, just like the example showed earlier, what is the causing certain outbreak and what can we do about it? Now, majority of a model is also used for a second purpose, which is to predict future. Ooh, that's so tricky. But basically what we are doing in here is actually more relevant to us in this case, looking at viability testing, is looking to what will be the impact, the potential impact on the population once we introduce, sorry, the slides just seemed already jump, when we introduce new products. And finally, we can also use modeling to evaluate scenario that sometimes is impractical or unethical to study empirically, which I probably will go through in some of the examples shown in this presentation. Now, the next question is, how do modeling can support industry? And to do this, I will just borrow a slide from Chinu, and I'll just go through it and do some new trick and explain it through modeling. So Chinu described 4 phases, and the first phase is to understand the current market. So in this case, modeling can be used to identify the causal relationship and mechanism, just like what we're doing in the outbreak model. And together with surveillance data, we can not only identify what is causing certain outbreak, for example, but also investigate what kind of interventions or strategy can be used to stop an outbreak, for example. Now in phase 2, and that is probably the mainstream of modeling, is to predict future. So we can use this model to predict what will happen if we introduce certain intervention or product in the population. Now for phase 3, we want to submit our regulatory submission of the regulatory body. And in terms of modeling, we allow us to provide some quantitative data in order to support the submission. And in the final phase, where everything is settled, then we want to look into the possible impact of introducing this certain product or intervention into the population. And we can use modeling to assess what happens if we introduce this intervention into expand the population scale in order to assess, for example, what kind of benefit and cost involved together with economic analysis, for example. And with that, that's my 3-slide introduction on modeling. Hope always understandable. So now I'm going to go to specific, in this case, look at the modeling of viability testing. So viability testing is, I think many of us would already familiar with the concept, so I will just keep the introduction brief. But essentially, antibiotic overuse has been known to contribute to the emergence and spread of AMR. Now, in most diagnostic tests we use, cannot distinguish between the viable bacteria, which is the bacteria that lead to transmission and need to be treated, as opposed to non-viable bacteria, which is the bacteria that doesn't cause disease or doesn't cause transmission, and therefore, technically, you don't really need to treat them. And because the diagnostic test doesn't separate those two, and therefore, there's a potential of over-treatment if we're just based on diagnostic tests alone. Now, a couple of us in the AMR Hub is also involved in the development of a point-of-care molecular test that is capable to separate viable and non-viable infection. And therefore, from the modeling point of view, what we want to do is, let's say we got this test available and ready to introduce the population. What would be the population impact once we introduce this into general STI diagnosis? And in this model, I'm particularly focused on the MSM population. Now, there's 2 main outcomes of this modeling. The first outcome is the impact of the antibiotic use, which is, in definition, is quantifying the reduction of overtreatment, which in this case, overtreatment is defined as the number of individuals who are provided treatment even without any viable infection on their body. Now, note that this definition is slightly different from the definition of how accurate, or it's more complex than how accurate is the viability test. Because there could be a situation, for example, where you have got a viability test that identifies non-viable infection in Thyrox, for example, but there is still a chance that treatment still need to be provided, given that they might have viable infection elsewhere in their body. Now, the second impact that we want to look into is the indirect impact of the risk of introducing viability testing. In this case, we are looking into the assess the potential of undertreatment, which means that potential undertreatment, which in here defined as a viable infection identified as non-viable due to limitation of a viability test. And as you can imagine, if that does occur, that could lead to non-treatment or undertreatment, which lead to a risk of further transmission. Now, obviously, we don't really want this to happen. So we will try to reduce this as much as possible. But modeling do allow us to assess what happens if that is unavoidable and what would be the impact on the population. Now, before I dive into the result, I just want to further clarify 2 concepts that is used in this study. So first is the sensitivity of the viability test against viable infection. Or this is quite parallel to the sensitivity of a typical STI diagnostic, where high sensitivity means that you identify, in this case, viable infection properly, and therefore can reduce undertreatment. And we'll be looking at the range of sensitivity of 95% or above. Now, the second concept is the sensitivity of a viability test on non-viable infection, which is closely parallel to the specificity of a diagnostic test, except that it's slightly different in concept. However, in both cases, high sensitivity in this measurement will reduce the number of overtreatment or unnecessary treatment on the population. And obviously, that's the thing that viability tests will aim to reduce. Now, having said that, because even though it's an ultimate aim, in the clinical setting, it's quite likely that that is a secondary priority when compared to undertreatment. So therefore, in this case, we will look into a range a bit wider. In this case, we are looking at sensitivity from 90% or above. Now, just like in many modeling study, the first step, and probably the most tricky step, most annoying step, is to establish a baseline scenario, or where you compare the scenarios from, or the status quo. So I'm not going to go through the number in details, but essentially this model will be informed by the incidence rate and the percentage of non-viable infection through diagnostic under the current situation. And then we calibrate the model, assuming that stay constant, and then generate scenario where we introduce viability testing and compare the difference between scenarios. Now, the first result I want to show is the main thing of viability testing, main objective of viability test, which is to reduce overtreatment. In this case, we're showing two graphs, one for gonorrhea and one for chlamydia. And the horizontal axis is number of years since the introduction of viability testing in STI diagnosis. And the vertical axis is the cumulative number of overtreatment under each of the scenario per 100 people. So under no viability test, it increased fairly linearly, the blue line here, which is not surprising because in the model, we do assume at baseline scenario is at equilibrium. So every year there's same number of infection and same number of non-viable infection full year. So it increased very linearly. Now, the more interesting part is to look at the impact of introducing non-viability tests under various level of sensitivity. So from 90%, 95, and 100. So you can see that in this case, by introducing a viability testing of this certain percentage, it will lead to from 88% to 100% of overtreatment prevented, which is equivalent to around 7.6 to 8.6 fewer overtreatment per 100 person years. Now, to put this number in perspective, the incidence of gonorrhea and chlamydia among MSM here is around 20 to 40 20 to 40 per 100 person years. So therefore, the number of overtreatment, when you compare it with that number, is relatively significant. Now, that's the good thing, but we also have to look at the possible risks as well of introducing viability testing. So in this case, we are looking into the case where the sensitivity of viability tests against viable infection can decrease from range from 95% to 100%. So with 95% means that there's a 5% chance a viable infection can be misidentified as non-viable and therefore not treated, and therefore can lead to further transmission and possibly lead to increase of prevalence. So in this case, we are looking the vertical axis is the relative change of the prevalence at the 5th year. So you can see in this result, if we got a viability test of 95% sensitivity, it has the potential to increase gonorrhea prevalence by around 6.5% after 5 years, which is something that we don't want to do. But it does allow, the modeling do allow to assess if undertreatment does happen, and then there will lead to certain level of increase, depends on how sensitive or how accurate it is. Now, interestingly, for chlamydia, the effect is actually quite negligible. And that could be due to the fact that even though they have similar incidence, the biology of chlamydia is quite different from gonorrhea, with chlamydia likely to last longer, less transmissible, and they don't have symptoms. Now, so far, in the previous result, we assume viability test has been used in every diagnostic, which is in reality probably doesn't make sense. Firstly, for example, it has been known that viability test is less sensitive, just like diagnostic test is less sensitive for oropharyngeal site. And therefore, it could be considered not using viability testing at that particular type of infection. And furthermore, if a person has symptoms, then you probably don't really need to apply viability test, given that chances are you will apply some form of treatment anyway. So with this restriction, then we can look into different strategy of viability testing. So for example, in this case, we are looking at the situation where we're using the same viability test with lesser sensitivity of viability against viable infection. but we are only using it on infection that's asymptomatic and urethral and rectal infection. And we're looking at gonorrhea again. Then in this situation, the potential impact of undertreatment is only around 1% increase of prevalence as opposed to 6.5% increase, which is still pretty high, but I guess it does reduce the risk of undertreatment. So in summary, the modeling allow us to assess the various viability test performance and implementation strategy that is sometimes not really practical to carry out. Our study has found that viability testing can prevent up to 8.6 overtreatment per 100 person years. However, if undertreatment does occur from viability testing, then it could increase STI prevalence, especially for gonorrhea. However, by choosing a proper strategy of using viability testing, such as focusing on non-oropharyngeal and asymptomatic infection, it could reduce the risk of undertreatment and hence better outcome. And with that, I shall conclude presentation. Thank you.
Thank you so much, Ben, for the presentation. I will go to the last presentations for today's sessions, and Nur Hayati Agus will be the persons that are presenting the next one. She's an epidemiologist and director of Yayasan Kasih Global Indo, leading sexual health clinics and research in HIV, hepatitis, STIs, and TB HIV in Indonesia. Your time.
Okay, thank you. Thank you, everyone. Please allow me to share the surveillance for antimicrobial resistant to gonorrhea using molecular testing in Indonesia. Next slide, please. So, into antimicrobial resistance into Indonesian context, the situation is at that time based on the integrated biological and behavioral surveillance in 2015, the gonorrhea prevalence is so high. It's like 1 out of 5 female sex worker has gonorrhea, and then 12 or almost 13% MSM and 12% of transgender. The gonorrhea prevalence the gonorrhea prevalence is positive. And then regarding the AMR challenge in Indonesia, based on the household survey in Indonesia, 80% of the household having self-medication at their home. And then resistant monitoring, this is conducted after my study about the molecular resistance for gonorrhea. They have a very small number and limited coverage, only 4 sentinel sites in Jakarta. And then we know that to understand the resistance for gonorrhea, we need to have a culture, and then this culture is very complex scale, and even though this is a gold standard, it is very complex, and then it's not robust for transportation. And then based on our experience also that for gonorrhea culture, we need to have the media that is not available in the country for commercially, and then that's why we need to make our own media for that one. And then until now, there is no published data on molecular resistance in Indonesia. Next slide, please. Based on the latest study in 2024, the resistant pattern in Indonesia is like 85% of the gonorrhea resistance with penicillin, and almost 80% with ciprofloxacin, 60% on tetracycline, and there's also showing the resistance in ceftriaxone 5.6%. This study is using the culture with the calibrated dichotomous susceptibility methods. Next slide, please. And then the study rationale for this molecular testing for antimicrobial gonorrhea is like the current treatment is cefixime for 100 milligram and azithromycin 1 gram. This medicine is being used since 2010. And then to provide the model for enhanced gonorrhea, Neisseria gonorrhea AMR surveillance in Indonesia. And then need for updated data on gonorrhea susceptibility using molecular. And this study will use the frozen sample from the integrated biological and behavioral survey. The national survey for key population for demographic and behavioral data, and including the clinical data on for key population. And then this result will support the SCI programming in terms of finding the new approach for the surveillance. Next slide, please. Okay. Now I'm going to say I'm going to share the method. So we use the cross-sectional study, the national big study for key population in 12 Indonesian cities. So we collect the behavior, and then we also collect a sample, blood sample for HIV and syphilis test, and also hepatitis. And then also we also collect the swab sample. This swab is using the urethral swab, anal swab, and also vaginal swab for a woman. And then we also use the sample from the Global Indo Sexual Health Clinic, the one who want to come to clinic with the gonorrhea positive in 2022. High risk population is included, and then oral informed consent obtained and confidentially ensured. And then the sample collection. The sample collection is a swab in PCR transport media for gonorrhea and chlamydia, and 327 positive samples are liquidated into a sterile vial, and then DNA provide using commercial kit and stored in minus 80 degree. And next slide, please. In this study, we use 6 primer and probe to screen the resistance. We use PPNG, penicillinase production Neisseria gonorrhea plasmid, to see the emerging of the penicillin resistant, and mosaic pen A for cephalosporin, FC428 for cephalosporin, GYR 91 for ciprofloxacin, 20 sRRNA 26111 for azithromycin, and then another 2059 for azithromycin. And all of this primer and probe is provided by the University of Queensland. Next slide, please. So this is how we are doing the resistance testing. So the storage sample from the IBBS and also the sample from the sexual health clinic, from their medium, and then we conduct the extraction using the DNA extraction kit, and then after several steps, we get the evaluate, and then after this we add the primer and probes So like I previously mentioned, there's 6 primer and probes. So each sample tested in 6 primer and probes, and then after that, going to the PCR process, and then we check for the internal control, and also, we also check for the positive controls that being provided, and then we check also the CT value to understand the resistance. Next slide, please. So this is to understand that there are 2 different population. Like for the IBBS is high risk population, female sex worker, transgender women, and men who have sex with men. While for the clinic population is the patient that attending the sexual health clinic for STI screening. And then the geographic coverage is in 12 cities in 7 provinces in Aceh, Jakarta, West Java, Yogyakarta, East Java, Bali, and East Kalimantan, while the clinic is only reach the greater Jakarta area. And then the biological sample is 2,837 for from the IBBS, while from the clinics only 50 respondents. And then all the sample tested using the PCR, if they are while for the clinic population is the respondent who are positive for microscopy and then offer for the PCR testing. And then after that we got 344 positive sample. However, for the IBBS, we can only test for 258 source sample from the original result 344, while for the clinic, we tested 49 sample because 1 sample is found out negative in the PCR. Next slide, please. So this is the population of this study from IBBS from 12,591 respondents. We exclude around 9,000 because we didn't collect the sample for chlamydia and gonorrhea. So for female sex worker, we tested 1,528, and then we got gonorrhea positive 168, and then from transgender, or we call it waria in Indonesia, we tested 514, and then we got 41 gonorrhea positive, and then for MSM, out of 795, we got 135 gonorrhea positive. And then all these positive sample, then we tested for the resistance of using the molecular, and then only 175 samples has a valid result. Because this is a source sample almost 4 years before the studied, so that's why there is some deteriorating, and that's why only 175 samples. And then while for the clinic, out of 385 clinic attendee, we exclude because only 5 clients have tested for positive for PCR gonorrhea, and then this out of 50, then 49 tested for resistance. Next slide, please. This is the characteristic of the respondent. For the age is almost similar. 52% below 25 from the IBBS and 47% below the 25 years from the clinic. However, in terms of education, in the IBBS is 51% has low education compared the one who come to the clinic, only 8%. And then for not married, In the IBBS is around 59%, and then in the clinic 86%. And then the difference is also in the employment status. Half of the respondent in the clinic is having the income below minimum wages of Indonesia. And then while in IBBS sample, around 89% has a minimum wages, has below minimum wages. And then now comparing to the STI symptom, because someone who come to clinic is already have the symptoms, so they have a higher symptom. And then regarding condom use, clinic has a lower condom use compared to the IBBS sample. Next slide, please. Now we come to the result of this initial screening of gonorrhea resistance. For the antibiotic of penicillin, around 84% from the IBBS sample, and then 71% from the clinic sample. We haven't found any resistance on the cephalosporin on the IBBS. However, we see the emerging cephalosporin for primer FC428. We found four sample is positive for resistance, and around 8% is resistance. And then for the ciprofloxacin is also similar, and then we haven't found any resistance yet in azithromycin. Next slide, please. So the conclusion is, this is the first study that in Indonesia that we use molecular methods. This is a culture-free for screening of gonorrhea resistance. And then learning from the tuberculosis, I could say that PCR can be a game changer because it's more sensitive, faster, applicable to all clinical samples compared to the conventional method. So I could say that learning from the tuberculosis, previously they could detect resistance only through the culture, takes 40 days. Now with the new point of care, and then there is also near point of care, they could detect resistance immediately. So I think from this we can learn that this could be also could be happen for the STI. And then the key finding is also the high resistance on penicillin and ciprofloxacin. And then we also already detect that after 4 years, there is a resistance on cephalosporin. Next slide, please. So the next step for gonorrhea resistant surveillance in Indonesia is we need to develop simple and affordable molecular method. So we can adapt this PCR into point of care testing, ensure that reagents availability, and make resistant testing more accessible. And then start using surveillance. So after this study, we engage with the government to start using surveillance, and then they start using the enhanced gonococcus surveillance in 2023. And then I think we need to conduct the longitudinal study to see on the impact of antibiotic self-medication, and then also establish sample referral system, because I know if we want to have like resistance in everywhere, it will be not economical and not efficient. So we need to establish the sample referral system to send the like we conduct for HIV. If we found something if we suspect there is a resistance, then we send a sample to the national reference laboratory, and then activated hospital reporting, because actually the hospital conduct regularly for the resistance resistant monitoring, and then from this, then if they are start to report it, then if we suspect there will be resistant in some antibiotic, then they could send a sample for referral. Next slide, please. So this is my last slide, and then we essentially thank you for the following partner for their support and funding that makes this study possible, to the AMR Hub, and then to Prof. John Caldor, who support the study, and Prof. Tanya from the Kirby Institute, and Dr. Kim Lowry and Professor David Wiley, and also Cameron from the CCR University of Queensland, Professor Pandi Januaraga from Yayasan Khatib Praja, and then our colleague from the Ministry of Health Indonesia, and then all of my friends from Yayasan Kasih Global Indo. Thank you. Over to you, Bia.
Thank you. Thank you so much, Nur Hayati. I think we are now, having some time for the questions, and we are happy to take any questions from the floor or from anyone online.
Is there anybody at the Kirby?
And if anyone's shy, you can also put it in the message box in the Zoom, and I'm happy to read it out loud as well for anyone online. Is there anyone from? I mean, maybe I can just start it because this is quite interesting for me, and I think while everyone's gathered their thoughts and maybe just thinking about the questions that they want to ask, I'm interested actually with the Nur Hayati presentation, especially because I'm Indonesian myself, with a very revamp of the diagnostic and laboratory in Indonesia, and all the ambitious thing that our minister is trying to do for the diagnostic and laboratory strengthening. Is there an opportunity to actually having something like a standard package to make sure that it becoming something that routinely to do in the laboratory diagnostic that are currently being developed, the lab CASMAS or what they call it as a public health laboratory? It is something that a window opportunity, you think, Nur Hayati, for resistant monitoring in Indonesia in a more general population?
Thank you, Bia, for your question. So, I know the ministry is very anxious to have a new technology and everything, and then also being supportive. However, so in terms of the system itself, the public health laboratory already has a set of system. If they found any sign of resistance, they could send the sample to the higher level. However, we know that the STIs is being neglected. I could say that because there's our EGAP study in 2023 is being supported through the Global Fund and WHO, and then STI still think that and gonorrhea is part of the STI is still not being monitored because it's related to the key population, it's related to the sexual transmitted. So and then we also know that there is no commercial reagents available actually for the resistance testing. So this is the window opportunity actually to because we learned from the TB that we could do it. So I think this is the window opportunity for STI to start the screening for the resistance using the sentinel sites, and then they could leverage the sentinel site with the biggest area in the one who has a very big positivity rate for gonorrhea, and then we can elaborate more site for resistance. I mean, elaborate more sentinel site to send the resistance sample. So actually, we already have the because at that time because there is public health laboratory is not established yet. So for the EGAPs, we use the Sulianti Saroso Hospital. Now we have to support the public health laboratory, the Balai Besar Laboratorium Biologi Kesehatan in Indonesia, the Tier 5 laboratory, so they could start to have this resistance as part of their responsibility. Over to you, Bia. Maybe
Thank you.
they
Thank
could.
you.
We have a
Yeah.
question on the Kirby.
Yep.
Hi, Nuriati, it's John Kaldor, and thanks, Rabia. Well done on your presentation, and thanks for joining us early in the morning from where I think you're in Saudi or Mecca or something like that. So I hope the trip's going well. Just to follow up Rabia's question, and following your response, what we really need to do is have a way of identifying gonorrhea cases quickly and widely, which is a project which Tanya has been trying to get up with funding, which we haven't quite got yet, in multiple sites around in Asian countries. But I think that's really the only way to start getting a more comprehensive primary care-based surveillance system, is to be able to target cases that are picked up through clinics that serve populations at high risk of gonorrhea, like key populations, and then quickly refer those cases that are detected using point-of-care testing to the few labs that can do that comprehensive testing. And until we have that, I think we're going to keep having this problem. But it's good that I think the sooner you can publish the data that you have from this study, the better to make the point that we're not really getting enough information on gonorrhea resistance from the very few clinical cases that present at the sites that are used. Maybe I just switch over to a different topic. Can I just ask a different question? Oh, sorry, I guess is Rabia chairing? Or you've both chairing. All right. Anyway, it's just going back to Ben's presentation and the viability testing. I don't know a lot about that technology, but I was wondering whether there's any update on when it might actually become field usable, or what's the pathway for implementation? Thanks.
Who from SpeedX wants to?
It's likely to be released officially at the end of the year as a research use only. IVD will be further down the line, but definitely as a research tool by the end of the year. But it is available for studies before that.
So do you have a pathway of strategic research that's going to take it forward? That's good. So and we're all collaborating in that, are we? Yeah, good to hear. Yeah, thanks.
I have a question for Qinglu. So I heard that resistance guided testing has similar costs but has benefits. So I'm guessing, how should that be communicated to make sure that gets implemented more often? Because I'm sure there's somebody who can comment on that in Kirby as well.
Hi, Andrew. Thanks for the comment. I think it's will be a positive sign that the resistant guided therapy costs similar to the resistant guided therapy. And I think some of the clinical guidelines have already put that in the guidelines. So it just maybe I think it's a matter of time to increase the uptake of this practice.
Okay. I think we are on time. Yes. Is there a question?
In the chat.
There is one in the chat,
Yep.
and I think it's from for Nur Hayati as well, from Fabian. I think Fabian asking about no AMR for azithro for gonorrhea, which is interesting as we see some AMR generally. Any comments on no AMR, Nur Hayati?
Okay, thank you. Yeah, because actually, the azithromycin is not directly for the treatment of gonorrhea. So we use azithromycin is for the treatment for because usually gonorrhea come with chlamydia, and then we use only 1 gram. So because we haven't used widely for like in other country use azithromycin for syphilis treatment. So uh, we haven't found any resistance yet in azithromycin. And then this is also compared to the previous study that's being tested, and also the newest EGAPs in. We haven't found any resistance yet in azithromycin.
I think that's really interesting because you can use it to treat gonorrhea. So you can use 2 grams to treat oral gonorrhea, which is much harder to treat. So I think it's really interesting that you can use this potentially in Indonesia. Thank you.
We have a Kirby question.
Yeah, on the note on azithromycin, the 23S mutation is only associated with really high MIC azithromycin resistant strains, so greater than 256, whereas clinical failure occurs in strains with MICs as low as 0.5 micrograms. So using that test to indicate resistance is not the full picture.
Right, gotcha. Yeah.
Can I just double check, was there MIC testing done as well, or were the calls based on the genes only?
For Indonesia, for this study, we only use the molecular biology because we use the source sample. However, for the EGAPs, we use the culture, maybe only limited, like I previously mentioned in Jakarta, in 4 sexual health clinic that has the biggest, and only for the urethral discharge, the sample. So maybe this is not representative because we know that the gonorrhea prevalence very high in female sex worker.
Any other questions? Nope. Give it back to you, Tanya.
Fabian, are you there?
I'm here, yes.
Is there something you'd like to say?
I would like to say something, yes. So I'd like to now present the winner of this year's EMCR Emerging Leaders Award. So we had lots of great applicants this year, high caliber research by EMCRs, and it's very impressive. The winner this year is Dr. Emma Sweeney from University of Queensland. And I just want to say a few words about her work. So Emma has made some significant impacts in the field of NG management, including contributing to clinical guidelines for CDC and in France. She's assisted in developing diagnostic tools at the Victorian Infectious Diseases Reference Lab using her work on NG biomarkers. She's had, obviously, the industry collaborations as a Ready Fellowship with SpearDex, where she led to a commercial diagnostic test for NG, and her leadership in mentoring and supervision of staff, postgraduate students, postdocs, and research assistants, as well as executive committee on on the editorial board of JSC Journal, and also earning great amounts of money of over $800,000 as a CI while she was on parental leave in 2021. So amazing work, Emma. Congratulations. And I think Ella Herring in the room is there with you. Ella, are you there? Ella has a beautiful bespoke certificate frame for you.
Thank you very much.
Yay! Congratulations. Thank you so much. You want to say anything, Emma?
Oh, no, thank you very much. It's nice to be, acknowledged for some of the hard work that I've done. So thank you to everyone.
I think the great thing about this symposium was it's really provided that balance between last year where we had that laboratory work and this year where we've also been able to focus a lot on all the other expertise that is equally important to be able to inform how these tests can come to market. I just really wanted to thank the speakers who have put a lot of effort into this. The presentations, we were discussing this with Ben and with Chinlu and others. No matter what you think you've got a presentation ready, you've done it before, it's never the same presentation, it's never the same audience. Nicole, you've for the first time presented something completely differently and did a fantastic job. So thank you to everyone for all the effort that they've put in. Also, thanks very much to Beck, who had the vision for bringing us all together under the AMR Hub and allowing all of this work to happen. making it possible. For the early mid-career researcher team, the committee, who have met in the background, I think once a month for the past, I don't know how many months, to pull it together. So there's a lot of work there. Thank you to Fabian, who had a very big whip, keeping us all very efficient with our making efficient decisions and getting our action items done. Michelle, Cass, Charisse and Vanessa in the background have also sort of been diligently working IT and making sure everyone's able to connect through these hubs, because I think it's not, speaking with our IT guy, Sergio, who was very patient with us this morning and provided amazing help and support. It's not a normal model to be able to have people in physically in many locations and yet still be able to join from the other side of the world in real time. So thank you to everyone behind the scenes to make this possible. And of course, to all of our partners who are listed here, many of whom have been able to join us today remotely as well. And again, we don't often get the opportunity to come together. So I just wanted to thank everyone for taking the time out of your busy schedules to spend time with us today, to share all of our knowledge and move this forward. So thank you, everyone.